All right, we are live, so transferring back to you, Catherine.
Great, and then I can make you co-host again. Okay, great. Lianne is here.
Okay, so
I've unmuted you, Lianne.
I'm ready.
I am tempted, Lianne, actually to not make you a co-host, because then that way you can raise your hand unless you prefer to be co-host and be able to just pop in. What is your?
I am happy to raise my hand.
Okay, why don't we do it that way then?
Cool.
All right, so I'm not on video, I see that. Here we are. Hi, everybody. Everybody can hear?
Can hear you.
Fantastic. Okay. So welcome back to day 2 of the 2020 Gellis Senior Symposium in Human Biology. And my name is Catherine Preston. I'm the Associate Director of the program. And to start things off, I'm going to quickly just kick things over to our director, Lianne Kurina, who's going to say a few words.
Thank you, Catherine. Hello, everybody. So good to see you all. And I'd like to start by congratulating the students, because I know what an enormous amount of work it has taken to get you to reach this academic milestone. So you should feel really proud of yourselves for this accomplishment. It's a big deal. Speaking of this accomplishment, would not have been possible without the guidance and leadership of Catherine Preston, who has facilitated this for all of you. So thank you, Catherine, for your leadership and hard work on that. Finally, it is my pleasure to introduce Jeff Gellis, who is here with us from New York City. And Jeff is a proud HumBio graduate from 1985 and has been a really important supporter of HumBio. So for him, his area of concentration was biological anthropology and cultural evolution. And he was lucky enough to be advised and mentored by Bill Durham, so one of our kind of key figures in human biology. So after finishing at Stanford, Jeff pursued an MBA at Harvard and has had a career in finance, and he now runs an investment management firm. But fortunately for us, human biology is still dear to his heart. And I was fortunate enough to meet Jeff just over a year ago, and was really amazed by his attachment to this program. So what you may not know is that a gift from Jeff helped us to establish the capstones in general. And now he's given a further gift to help support the senior symposium. So a lot of dedication to our HumBio students. So with no further ado, thank you, Jeff, and I'll hand it over to you.
Thank you, and thank you for your kind comments, and really appreciate it. Look, I won't take long. I just want to make, I don't know, 3 points. Before that, though, I am in New York, but I have to say, and I won't add any commentary, but this is Hong Kong behind me. Just saying, Hong Kong. Support. At any rate, so 3 quick comments. One, yeah, I graduated in '85. I mean, you know, this was going to be my 35th reunion year, which is really daunting in a sense, but you know, it's kind of cliche, but it happens fast. For those who were on yesterday, I apologize if I'm repeating a few things, but look, it's remarkable to think back over that time period, but it does feel like I'm still in the room, in the core, and I loved it then, I love it still. I'm a huge supporter of human biology, and Lianne and Catherine and the team have been fantastic. I view it as a worldview, a perspective that I carry with me every day. I hope you will, and I'm sure you will as well. And a plug is, it is the best department on campus. It is the place to be. And, you know, I really do appreciate having been part of the capstone project or effort and part of this as well. The point I would just make is that, you know, of all time to be a HumBio graduate, in the world that we're in right now, you know, if there's ever been a time in history that, you know, the A side and B side have come together better and more importantly, to bring that view, to bring the biological and the social science perspective together, the demand and the need is so great. And I know it's been hard and the hardships placed on you as seniors. And, you know, I really, you know, feel for each of you and the impact it's had. But the need in the world for the perspective and the education and the background that you have, the need is running as high as it could be right now. So it's kind of exciting in a sense, even though it's, you know, not for the better, but really what a time to be bringing HumBio into the world. And then finally, just really a note of congratulations. Tremendous achievement, honors, HumBio, Stanford. I mean, congratulations to each and every one of you. You will always have a home in HumBio. I return to the department every time I'm on campus, and I really, you know, welcome you to do the same. You will have a home in HumBio forever. And just a final note, if I could offer any 2 cents of advice, it would just be pursue your interests and good things will come. Pursue your interests and good things will come. So with that, I'll hand it back to Catherine. Enjoy, have fun, congrats, and have a good day.
Thank you so much, Jeff. And yeah, I want to echo what he said about how important HumBio and the whole HumBio approach is right now. And I think that throughout the rest of the afternoon, people who are listening to our students talk are going to see just the amazing diversity of approaches that people have taken to problems. Every single one of our students is addressing a problem that is so important to humans and the planet right now, and everybody has different ways of approaching it. That actually reminds me to mention that we have a mix of different projects. We've got kind of standard traditional honors research projects, but we also have sprinkled throughout the day senior synthesis projects. And one of the things that I think is so cool about the senior synthesis is an opportunity for students to do something that is really in depth, that really grapples with a problem, but it's not traditional research per se. And I've been able to try to match some of those that and I think that's just another example of how you can use a different approach to thinking about problems. And we're going to have the end of this first session includes a senior synthesis project, which is going to be really cool. Okay, so really quickly before I start the first speaker, just to let people know, there should be a participants panel on the right side of your screen if you're on Zoom. If you don't see it, you can kind of mouse down to the bottom and just hit participants and it will show up. If you're a participant, you will have a little raise your hand, a little blue hand at the bottom. So while some of our presentations are pre-recorded, the students are here to give live Q&A. Just raise your hand and I will recognize you and unmute you. All right. So without further ado, I'm going to things off with Jessica Welch.
Hi, my name is Jessica Welch, and today I will be talking about successful aging and lifestyle behaviors, integrated models for predicting the integrity of white matter tracks in older adults with mild cognitive impairment. Here's an overview of the things I'll talk about today. First, let's begin with the background. Successful aging, what is it? Successful aging is a concept that takes into account both positive and negative influences that define and modify the aging process. For this study, I focused on the framework put forth by Rowe and Kahn, and it posits that individuals who age successfully have increased rates from freedom from disease and disability, maintain high cognitive and physical functioning, and have high social engagement. The next term to define is white matter. White matter is made up of the myelinated axons in the brain. Think of them like highways that carry signals to and from various regions. We can measure the integrity of these tracks in several ways. For this study, I have chosen to use fractional anisotropy and mean diffusivity, which are ways to quantify organization of the track and degrees of myelination. These are collected through diffusion-weighted imaging scan, which is an MRI-based integrity. Diffusion-weighted imaging is a fairly new technology, and researchers are just beginning to expand their knowledge of the function of each individual track. Many tracks are not completely independent from one another in their function. For example, there are a few tracks that are involved in different aspects of memory function. Many current studies focus on understanding dysfunctions of white matter tracks and understand how they relate to various conditions. However, what has not been studied is evaluating white matter track integrity in healthy individuals as well as individuals with preclinical dementia. In order to fill this gap, I focused on a disorder called mild cognitive impairment, a disorder characterized by cognitive decline that is more severe than expected due to age and may affect memory, language, thinking, and judgment. Individuals with MCI can still maintain an independent lifestyle, and it's estimated that anywhere from 30 to 50% of individuals with MCI may go on to receive an Alzheimer's diagnosis. So understanding structural connectivity at this stage, as well as factors that affect aging, could be key in understanding possible interventions to delay impairment in the future. My research questions were as follows. 1, do the combined influences of the factors of successful aging better predict white matter integrity than any of the individual factors in isolation? And 2, how do lifestyle factors related to successful aging correlate with white matter integrity in individuals with MCI versus healthy control? Now here's a brief overview of the study design. Study visits and an at-home questionnaire helps us gather information on cognitive ability and other factors that fall under those domains of successful aging mentioned previously. And here's a brief look at the participant demographics. We enrolled 38 MCI participants and 22 healthy control, and the age range was anywhere from 65 to 85 years old, with the average age at around 73 years old. Now I'll talk about the analyses I did and their results. So first, I created models that reflect the individual categories of successful aging. These models look at the participants regardless of whether they're MCI or healthy control. The first model was emotional and psychological factors, which included factors such as attitudes towards aging, resilience, and optimism. Now, it's not important to focus too much on the details of each of these graphs, but to see that the higher scores of emotional well-being are associated with higher FA values and lower MD values, both of which indicate higher structural integrity. The next model was cognition, and you can see similar trends. However, there were only significant associations in 2 of the tracks. Model 3 included metrics of physical health, including exercise levels and history of disease. This had similar trends to the other models, however, had significant associations in 5 tracks. Model 4 was metrics related to social support and engagement, and after controlling for covariates, there were no significant associations. The takeaway from this data is that the individual aspects of successful aging are associated with higher structural integrity in some of the white matter tracks. Next, I wanted to combine the factors of successful aging to understand how a more integrated definition of aging correlated to white matter tracks. The model first combined the first 3 aspects of successful aging: cognition, emotional well-being, and physical health. And as you can see, higher scores in this model also predict higher FA values and lower MD values. However, this model had significant associations with 9 different track values, supporting my hypothesis that the combined influences of factors of successful aging are more predictive of connectivity in the brain than any of the individual factors alone. Additionally, when looking at which tracks were predicted by the individual and combined models, one important trend was clear. Many tracks that were seen in the individual models also appeared in the combined models. However, there were 2 tracks that were only significantly associated with successful aging when the factors were combined. These were the cingulate and the cingulum hippocampus. They're pictured here and are both tracks implicated in memory and cognitive control, two key factors of cognition that many individuals with MCI struggle with. This indicates that individual factors such as cognition, physical health, or emotion on their own are not sufficient definitions of aging, but together may better reflect the trajectory of functional decline. For this next set of analyses, I wanted to compare MCI to healthy control. To do this, I created a new model that includes factors of lifestyles that have been associated with positive outcomes in aging that go beyond the definition of successful aging to include factors such as cognitively engaging activities that have been proposed to affect structural integrity. Removing cognition from this model helps us understand differences between groups without the influence of cognition that we already know exists between them. When comparing lifestyle factors to FA values, we see increased positive lifestyle factors on the x-axis are positively associated with FA values on the y-axis for healthy controls, which is in blue. However, for MCI in green, we actually start to see decrease in FA values. We see similar trends when looking at MD values. For healthy control in purple, higher lifestyle scores are associated with decrease in MD. But in MCI, in orange, we actually start to see an increase. From these results, we can conclude that the relationship regarding white matter tract integrity in individuals with MCI is more complex than that of healthy control, and the individuals with MCI may have differing degrees of neuropathology associated with genetic risk factors of dementia. To evaluate the presence of neuropathology in these MCI participants, I use a previous model, but this time controlled for cognition. The outcome of this model shows that there's more severe neuropathology than the individual's presentation of symptoms of cognitive decline. This indicates that there is some mechanism of compensation at work that may be delaying the onset of clinical symptoms. One hypothesis is that these factors of lifestyle could contribute to building up cognitive reserve, a concept that proposes that more neuronal resources can help confer resilience against neuropathology in individuals who may have a higher risk for developing Alzheimer's, and this is illustrated here on the figure to the right. Lastly, here are some final conclusions. One, individual factors of successful aging may be able to predict white matter integrity, but a more holistic model of aging should be used when defining functional decline. And two, lifestyle factors predict higher structural integrity in healthy controls, but not MCI. And lastly, individuals 65 and older is the fastest growing demographic, and research into factors that affect the aging process is imperative right now. People diagnosed with MCI are at a higher risk for developing Alzheimer's and are a population that would benefit the most from research into protective factors and possible interventions that may help them maintain cognitive function and be able to live independent lifestyles. I want to say a special thank you to Dr. Hadi Hosseini, my C-Brain lab coworkers, and especially my fellow HumBio classmates. Thank you.
Okay, Lianne, already there's a question. Where is she? There she is.
Great, thank you. Great presentation, Jessica. It sounds like you thought about this really hard. Here's my question to you. Do you have a sense of the causal direction here, right? So we might like to hope that lifestyle factors and these others in your kind of terms predict the structural integrity, but I couldn't help wondering if exactly the reverse process might be happening as well. That's all.
Right. So for a lot of the individual factors, the causal relationship has been looked at. So a lot of, for physical health especially, they've looked at, lifetime history and how that can predict like long-term outcomes. But for my study, looking at the bigger picture helped understand the interaction for a lot of these terms. So for right now, I can't say whether people who have better lifestyles, that's helping them in the long term, or people who have better cognition by default have these. So individually, those have been investigated, but hopefully in the future, more iterations of the study could be used to then look at the causal relationship of all the combined factors.
Paul.
So nice job, Jessica. So Lianne was kind of going where I was, but I'd go one further. So to try to unravel this, would you want to make a larger cross-sectional study like you're doing? Would you rather do a larger cohort or prospective study? What do you think? Because it's really intriguing. It's going to be very hard to know, sort of chicken egg kind of thing here.
Right. I think the next step is first, as you said, getting a larger cohort, but also looking at different demographics. So a lot of the participants we had really high levels of education and kind of had a narrow focus. They were all from the Bay Area and had high occupational attainment. So I think the next step would be looking at a broader range to maybe see how different backgrounds and different levels of education might affect these. And then you could get a better idea for kind of next steps after that.
Okay, fantastic. Thank you so much. We're going to move on to our next speaker, who is Jacob Shaw.
Hi, everyone. My name is Jacob. I'm going to be presenting to you today about the effect of baseline performance and age on improvements in cognitive training regimens in older adults. So my thesis is based on cognitive training, which is a set of guided mental activities that past research suggests may be relevant in delaying the onset of dementia and Alzheimer's. However, individuals vary greatly with respect to their cognitive training outcomes, making it especially important to consider individual differences, in particular baseline performance and age, when understanding who improves most from cognitive training. And two past theories have sought to investigate this phenomenon. The first is the compensation effect, which states that older, lower baseline ability subjects improve most from cognitive training because they have the most room for improvement. On the other hand, the magnification effect hypothesizes that younger individuals with higher baseline profiles improve most from cognitive training because they have the most efficient resources from which to draw on. So with this background knowledge in mind, I conducted a literature review with my lab that summarizes the existing conditions, existing literature to contrast the conditions supporting the magnification and compensation effects in order to better understand how age and baseline performance impact cognitive training effectiveness and how that varies across three unique cognitive domains: episodic memory, executive functioning, and working memory. And so what we found in study one was quite convincing. We found that in the domain of episodic memory, which has to do with the memory of people, events, facts, what we found was that 5 out of 5 studies supported the occurrence of a magnification effect and the idea that younger, higher baseline performance individuals improved most. On the other hand, in working memory cognitive training, working memory having to do with more short-term memory, we found more evidence for compensation effects and the idea that lower baseline performance individuals will improve most from cognitive training. And so we see here that the effect of individual differences on cognitive training tends to be domain specific. And so with that research in mind, we drew a few conclusions. First was that given that episodic memory is the first cognitive domain to suffer with aging, it is less amenable to improvements in older and lower baseline profile individuals. On the other hand, in working memory training, given that the disuse hypothesis explains how lack of practice in working memory leads to suboptimal use of neural resources, these resources can be reactivated through adaptive cognitive training in working memory, which is why we see the increased improvements in lower baseline profile individuals. And overall, there was a lack of past research in processing speed and executive functioning cognitive training. And so that led me to study 2, which was the crux of my thesis. And in study 2, we took individuals aged 65 years and older and assigned them to 2 different groups. The first group was the cognitive strength group. These individuals were assigned to a cognitive training regimen designed to improve a domain in which they were proficient at. On the other hand, we had the cognitive weakness group, which was assigned to a cognitive training regimen designed to improve a cognitive weakness. And we wanted to follow these 2 groups over the course of the study to try to understand which group was going to derive the most training-related benefits from their cognitive training regimen in 3 unique cognitive domains. And those domains were inhibitory control, processing speed, and episodic memory. And this cognitive training was performed on the online platform of Brain HQ. And moving on to the results, what we found was actually quite convincing. Across many exercises in the NIH toolbox, which is a battery designed to test different aspects of each individual's cognition, we found very sizable effect sizes across all participants and all training groups And particularly, I wanted to draw your attention to the cog fluid comp metric, which is here on the left. And what we find with this metric is that across all participants, we see improvement. And this is meant to represent fluid intelligence, representing the idea that we are seeing improvements in an individual's ability to solve novel problems, which is especially important in older age. And moving on to individual differences, we see enhanced cognitive training improvements in lower baseline profile individuals across all participants. Look on the left here, we see a negative correlation between cognitive fluid comp at baseline on the x-axis, which is baseline cognitive fluid ability, and improvements. On the right here, we see a positive correlation between age and cognitive training improvements on the y-axis. So these two correlations suggest differentially, or they reinforce the same idea, which is that lower baseline performance individuals, those who need the improvements the most, are improving most from training. Moving on to the processing speed group, we see a similar story. We do see that the lower baseline performance individuals are improving most. With the graph here on the right, we see that the cognitive weakness group is improving significantly more than the cognitive strength group in processing speed cognitive training. And the same story here was evident in the NIH toolbox. We see across a variety of cognitive domains, including processing speed, working memory, fluid intelligence, we're seeing enhanced improvements in the cognitive weakness group, suggesting that PS, processing speed cognitive training, is especially amenable to low baseline performance individuals. Moving on to IC, cognitive training, inhibitory control, the effect of cognitive training was less clear-cut. However, we did see more evidence for compensation effect, as observed on the left here with the flank inhibitory control task and a ray auditory learning episodic memory task. However, we also saw a select task that favored the cognitive strength group and episodic memory, suggesting that in the inhibitory control group, efficient resources were necessary to derive improvements in episodic memory. And finally, in episodic memory group, we saw increased improvements in the cognitive weakness group in episodic memory focused tasks, which was in contrast to study one, where we only found enhanced improvements in high baseline performance individuals. But we also saw in the domain of executive functioning, enhanced improvements in high functioning individuals in the cognitive strength group, suggesting differential improvements depending on your cognitive profile in after episodic memory cognitive training. And so to wrap up, I wanted to draw a few conclusions for everyone. The first is that cognitive training is effective at inducing improvements in a variety of tasks in all training groups, especially in low baseline performance individuals. And this held true in the processing speed group and the inhibitory control group. In the episodic memory group, we see differential improvements in both low and high baseline performance individuals. But overall, we see that the individuals who need to improve cognitive functioning the most are deriving the most benefits from our cognitive training regimen. And in general, our findings help to paint a more complete picture of how we can personalize cognitive training to maximize benefits attained in each individual and help to slow down the cognitive aging process. And I wanted to also thank Hadi Hosseini, my mentor, David Lyons, my advisor, and the C-Brain Lab for all their support through this project. Thank you all for listening.
Great job. Okay, we've got 2 hands up and both of them need to be quick. So I'm going to start with Annette.
Hi, nice presentation, Jacob. I wondered if you could tell us a little more about what the cog fluid comp training actually entails. So what kinds of activities were people actually doing?
Yeah, it's a great question. So the cog fluid comp was a component of the NIH toolbox, which was completed before and after training. And it's a compilation of many different tasks that are meant to represent one's ability to solve novel problems and react to information quickly. And so what that finding was showing was that through a training focused in one specific cognitive domain, we're seeing generalizable improvements to fluid intelligence, which is kind of which was not being trained in particular, but was actually obviously a component of some of the tasks in which individuals were training. So that was a very encouraging finding, because that's ultimately one of the most important indicators of training effectiveness outside the training arena.
Thanks.
Great question.
Okay, I'm going to go to Anya.
Hi. Oh gosh. So I was wondering, Jacob, what was the most surprising part of your findings?
That's a great question. I would say I was very surprised to see that we could actually derive these enhancement benefits in the individuals who are lower profile, because previously I kind of thought that it would require individuals who were already functioning at a high level in order to really benefit from these type of intensive activities. But I was very surprised and encouraged to see that perhaps with these findings, we might be able to generalize this training to a subject body with maybe mild cognitive impairment, individuals who are even lower functioning, but could really use this training. So that was a surprising finding. Thanks.
Okay, that is fantastic. I want to keep us on track time in terms of time. So we're just going to move on to our next speaker. Are you ready?
Well, yeah, give me 1 second while I set this up, and then I'll be able to share my screen.
Yeah, just share your screen.
Okay, once that loads. Can everyone see that? Can everyone hear me first of all?
Yes.
Okay, sweet. All right, I will go ahead and get started. All right, hi everyone. My name is Artenisa Kulla, and I will be presenting my thesis on inflammation and frontal limbic connectivity in adolescent depression. So to give an overview of my talk, I'm just going to start with an introduction to sort of put the motivation behind the project into context. And then I'll go with an overview of the methods and the 3 hypotheses that we had, an analysis of each of those 3 hypotheses, and then end with a takeaway and some acknowledgements. So major depressive disorder is one of the leading causes of disability worldwide, and it is a psychopathology that often emerges first in adolescence. It is characterized by heightened inflammation precipitated by psychosocial stress and altered connectivity between the amygdala and the ventromedial prefrontal cortex. So these are 2 regions in the brain that are implicated in emotion regulation, and this is relevant to depression. However, few studies have examined the association between inflammation as measured by cytokines, which effectively are just proteins involved in the immune system, and amygdala VMPFC connectivity in depressed adolescents. So to give an overview of the methods, we had 114 participants recruited from the community, 52 of whom were diagnosed with major depressive disorder, and 62 were healthy controls. So each participant completed a resting state functional magnetic resonance imaging scan and a diffusion weighted MRI scan. So from the resting state fMRI scan, we were able to compute amygdala VMPFC functional connectivity. So we can see the VMPFC here and the amygdala here. So functional connectivity refers to the temporal dependency between 2 brain regions, and it reflects the level of functional communication between those 2 regions. So how much or how little are these 2 brain regions talking to each other? In addition to functional connectivity, we were able to compute structural connectivity, which can be measured non-invasively using diffusion-weighted imaging, and that's sensitive to the diffusion of water. And so it produces a 3D image of the brain as a function of that diffusion. And so we used tractography methods to segment the uncinate fasciculus, which you can see here. And so this is a white matter tract that connects the VMPFC and the amygdala. And from that, we were able to compute fractional anisotropy, which effectively is a diffusivity metric, and it's the directional preference of diffusion. So it reflects white matter organization of tracts. And finally, we were able to assay concentrations of interleukin-6 and tumor necrosis alpha. Those are the cytokines I mentioned from dried blood spots using a Luminex multiplex array. Okay, so the 3 hypotheses that we had were that depressed adolescents would have higher levels of pro-inflammatory cytokines, they would have lower amygdala VMPFC functional and structural connectivity, and that higher levels of pro-inflammatory cytokines will be associated with lower amygdala VMPFC functional and structural connectivity in the depressed adolescents. So the first analysis that we had was looking at inflammatory markers and depression. So using linear regression, we examined differences in inflammatory markers between the 2 diagnostic groups. And we used universal covariates, including age, gender, body mass index, antidepressant and antidepressant medication use. So as we hypothesized, we found that the depressed adolescents had higher levels of both IL-6 and TNF-alpha than the non-depressed adolescents. And these findings are particularly interesting and worth highlighting because they highlight the robustness of inflammation as a marker of depression. And it extends previous literature that we've seen in adults in which inflammation plays a role in precipitating depression. And now we see that also in adolescents. So then the second analysis that we did was running functional connectivity and depression. So unfortunately, we did not find any group differences in amygdala, VMPFC, resting state functional connectivity. But if we limited our covariates only to age, gender, and BMI, we did find that the MDD group had significantly lower left amygdala VMPFC functional connectivity than the control group did. And this agrees with previous literature. So this non-significant finding can really just be due to lack of statistical power, and that with a larger sample size, we would see these group differences play out. So then we looked at structural connectivity. And again, we tested whether there were group differences in diffusivity metrics averaged along the entire tract of the uncinate fasciculus. And using a linear regression with those universal covariates, again, we didn't find any significant group differences. But when you control for motion during the diffusion weighted scan and corticosteroid use, we did find that the MDD group had higher FA in both the right and the left UF. All right, so given the evidence that the UF is one of the last major tracts to mature, which essentially just means attain peak levels of myelination, the higher FA of the UF observed in the MDD group may actually reflect accelerated maturation of the white matter in adolescent depression. Then our final analysis was looking at functional connectivity and inflammation. So I'm only showing one analysis here, but this was run across both right and left amygdala and IL-6 and TNF-alpha, but unfortunately we did not find any significant associations between inflammation and resting state functional connectivity in our sample. This literature is pretty scant. Very few studies are actually looking at amygdala VMPFC connectivity, let alone looking specifically at functional connectivity. So again, in our sample, we just didn't find anything significant. But when we looked at structural connectivity, we did find that higher levels of TNF-alpha were associated with reduced FA in the UF, even when we're controlling for the diagnostic group. So higher FA values are thought to reflect increased structural organization of white matter tracts. So in our observed pattern of lower FA in relation to higher levels of inflammation, this might actually indicate demyelination. So that was probably a lot, but to put this all together, while we did not see any direct associations between inflammation and functional connectivity of the amygdala VMPFC, it could be the case that downstream effects of inflammation are affecting functional changes in the adolescent brain via direct effects on brain structure. So the model that we posit here is that general inflammation is driving these accelerated aging processes, as we saw in analysis 2, that eventually lead to demyelination earlier than we would typically expect. And then that eventually leads to function, so a change in the function and emotion regulation neural circuitry, and potentially explaining a lot of the functional effects that we see in adolescent depression. Okay, so that is all that I have for my presentation. I would just like to thank pretty much all of the members of the Stanford Neurodevelopment Affect and Psychopathology Lab, most specifically my mentor, Dr. Tiffany Ho. She's just been incredibly patient and has taught me so much throughout this whole process. Of course, my PI, Dr. Ian Gottlieb, and my second reader, Dr. Lisa Medoff, have just provided so much guidance and support. And of course, the participants and their families for contributing to the research. Cool. Any questions? I'm gonna stop sharing. Cool.
Thank you. That was great. So I'm looking for hands up. There's a little delay. Oh, and here we go.
Oh, great presentation, Artenisa, really interesting. I'm trying to understand the causal model. So is it your proposal that that these inflammatory changes predisposed to depression, or is it the case that onset of depression, which I could imagine actually precipitates these changes in inflammatory factors, which I didn't quite get that pathway straight.
Yeah, so it's a little complicated, right? It could be both ways. And I think that's what makes this sort of research / this area of research so interesting, and that it's kind of difficult to parse out exactly what's going on. So stress is a precipitant. They've shown this in animal models. Well, they'll inject, I can't remember what the exact thing is, but essentially an inflammatory marker, and then they will see depressive symptoms in animals. So it could very much be that causal effect of, okay, the inflammation is causing this. But it could also be the other way around, where perhaps because you are depressed, then it's leading to this other downstream effect, sort of stress that you have in your life, and then you see these inflammatory markers. So again, I think it goes both ways, but it's kind of difficult to parse that out, especially in humans, because we can't really dig into their brain to see what's going on.
All right, we have a question from Jamie.
Hi, so that was great. It's kind of taking off in the last question a bit, but I was wondering, do you think that these inflammatory markers are acting actually as inflammatory markers in the system, or do you think that they could be acting as other kinds of modulators of neural activity?
Well, I think they cross the blood-brain barrier, so they could be acting as both. They could be acting as signaling mechanisms, or they could be acting directly as, effectors. So perhaps both, but I'm not sure.
Okay, fantastic. Thank you. All right, we are going to move on to the next speaker, Andrea. I'm going to share my screen.
Hello everyone, thank you for listening to my presentation. My name is Andrea Banuet, and my thesis is on diagnostic error disparities by age, gender, and race. I will start with a brief introduction into my topic and then move on to my research question, methods, and my results. A diagnostic error is any failure to establish an accurate and timely explanation of a patient's health problem, or a failure to communicate that explanation to the patient. Diagnostic errors result in 44,000 to 98,000 deaths and 4 million serious injuries every year in the United States, making this a huge problem within our healthcare system. Interviews, studies, and media publications suggest that 3 groups in particular are more vulnerable to diagnostic errors: women, African Americans, and young people. Yet the dearth of literature on this topic makes it hard to assess the problem and create targeted interventions. I sought to fill some of the gaps in the literature by investigating how being female, young, and/or African American contributes to the diagnostic process. I did this by focusing on individual patient perspectives and national data on medical malpractice. My research took on a 2-pronged approach. In the first phase, I conducted patient interviews. The interviews included the experiences of 4 patients who identified as female, 2 as African American, and 5 as young. With narrative and thematic content analysis, I constructed a framework for how diagnostic errors unfold and identified patient-perceived biases against these demographics during the diagnostic process. In the second phase, I analyzed national data on medical malpractice using the National Practitioners Data Bank, and I compared this data to healthcare utilization figures provided by the CDC. I used these sources to characterize the frequency and the risk of diagnostic error by gender and age. Unfortunately, race was excluded from the second part of the study because it is not reported in malpractice claims. From the interviews, I found that diagnostic errors followed consistent patterns. The general process included 4 key events. First, an early medical encounter where a patient engaged with the medical system and the medical provider gathered insufficient information, interpreted the information incorrectly, or failed to evaluate that information within the context of the patient's medical history. The second event involved worsening or persistent symptoms. After trying to cope with worsening symptoms, patients re-engaged their providers, who reinforced previous diagnoses and asked patients to just wait it out. This usually led to mounting concern, denial, and confusion, where patients continued to experience a health problem in addition to added stress, worry, and self-doubt. It was at this stage that providers were more likely to run additional tests or consult colleagues about the situation. The last stage involves the aftermath of a diagnostic error. Even after a correct diagnosis is made, patients have to deal with the advanced disease, the effects of a wrongly administered medication, depression, and/or anxiety from their experience. In addition to this conceptual model for diagnostic errors, the interview responses yielded a set of overarching themes that highlight patient-perceived sources of error due to inappropriate evaluations of a patient's visible characteristics. They are presented here by the characteristics they apply to. In short, younger patients perceived that because they were too young to have anything serious, doctors ignored critical signs and emerging evidence and fixated on milder, incorrect diagnoses. Female interviewees perceived that providers did not take their complaints seriously and their symptoms were commonly trivialized and attributed to stress or hormones. African American patients similarly thought that doctors did not spend enough time on their cases and they felt a lack of agency in their diagnostic process. Overall, these patient perspectives validate that being female, young, and/or African American unduly impacts the diagnostic process through these mechanisms. For the second part of the study, I pivoted from individual perspectives to nationwide data. I analyzed over 91,000 malpractice claims, 35% of which were diagnosis-related. On this slide, you can see the distribution of diagnostic error by gender and age. 52% of diagnosis-related claims involve female patients, and 84% involve patients under the age of 65. These proportions match the distribution of the United States population as a whole, initially suggesting no disparities. However, in order to assess who is at higher risk of diagnostic error, we couldn't just compare malpractice claims to the whole US population, because not everyone in the US is equally exposed to the healthcare system. For example, the elderly tend to go to the doctor more, so intuitively they would be more at risk of diagnostic error. When analyzing the risk of diagnostic error by rates of exposure to the medical system, we found some unexpected results. First, being female does not increase your risk of diagnostic error. In fact, it lowers it by 23%. In contrast, being young nearly doubled the risk of diagnostic error, a much higher estimate than most research publications suggest. The one exception to this was patients who were 18 years of age or younger. Like female patients, this group was less at risk for diagnostic error. Specifically, they were 8% less likely to be involved in a claim relative to patients who were 65 and older. The decreased risk of diagnostic error for females and patients under the age of 18 is unexpected. However, it is important to consider the many social and economic barriers to filing a malpractice claim. With these considerations in mind, it is easier to see why women and children are less at risk for diagnostic error, since this is being measured partially on the ability to file a claim. In conclusion, patient perspectives indicate that being female, young, and/or African American unduly impacts the diagnostic process in concrete and consistent ways. And malpractice data suggests that while females are not at higher risk for diagnostic error, younger patients are. These findings can help healthcare providers improve clinical judgment and address institutional shortcomings related to the diagnostic process and care of all demographics. Thank you.
That's great. Thank you. We already have a question from Jamie.
Nope. Sorry.
Oh, sorry. I guess
Yep.
I never lowered your hand after last time. So Lianne, I'm having trouble there we go. Are you with us, Lianne? Lianne.
Try again. Okay, here we are. Good, thank you. Great presentation, Andrea, thank you. How did you identify the people with whom you spoke? I guess is question number one. And how do we know that they're expressing, how do we know that their expressions of their experience differ from people who would identify as old, male, etc.?
Yeah, those are great questions. So we had a set of inclusion criteria for which we selected participants from. They had to self-identify as being female, young, and or African American, in addition to having had an experience where they felt that those visible characteristics impacted their diagnostic process. And after that, after these participants were selected, we conducted the semi-structured interviews. And I think you're right, it's very important to kind of compare them to other demographics to see what these disparities are. The study is ongoing, so we'll definitely have other controls we'll compare them to. But based on the pool of participants that we did interview, we did find there were significant disparities in how patients with these demographics specifically perceived that their diagnostic process was influenced compared to older patients, white patients, and male patients.
Great, and our next question is from Jennifer.
Thank you. I had some more specific questions along the line of what Lianne was asking. Can you share a little bit about how much time had elapsed between the time that you and your colleagues conducted the interviews and the time of malpractice? And can you talk a little bit about how you mediated that in the interview process?
Yeah, for sure. So there was kind of arranged a little bit. Some participants had very recently had an experience with diagnostic error, while others it had spent a couple years from when their diagnostic experience happened. Not all of them resulted from the interviews, not all of them resulted in malpractice claims. But we definitely tried to mitigate some recall bias by obviously not asking leading questions and just asking participants to share from their experiences and provide evidence whenever possible about medical records or instances where they specifically felt that these characteristics tangibly impacted their processes.
Okay, fantastic. Thank you.
It seems like that would be especially tricky. Oops, sorry.
Sorry, Jennifer, can you repeat?
Oh, sorry, I was just following up to say it seems like that would be especially complicated dealing with young people. Thank you for your answer.
Yeah, thank you.
Terrific, thank you. So we're now going to move to Shravya Gurrapu, who is one of our senior synthesis students, and I'm just going to turn it over to her.
Thank you. Give me one quick second to share my screen. Awesome. So as Captain introduced me, my name is Shravya Gurrapu, and I'm a senior who's doing a synthesis project on understanding underrepresented South Asian identities and narratives through Kuchipudi dance. So to start off, I would like to talk to you a little bit about Kuchipudi dance, which is an ancient South Indian art form that I've been learning for over the last 15 years. And this art form is deeply rooted in both cultural and religious traditions and has a lot of symbolic meaning and gestures, all the way from the dancers' foot movements and postures, even up to their eyebrow movements and eyebrow gestures. And from a very young age, dancers are trained to sort of understand what each of these symbolisms mean and how to utilize them to portray a different story or narrative. And since coming to Stanford, I focused a lot of my HumBio education on sort of understanding the cultural and social implications for health and health disparities. And something that I started to realize was in my own portrayals of Kuchipudi dance, the ways in which I myself had been trained to portray images such as femininity and masculinity were very different and carried with them a lot of very heavy connotations that I had never considered prior. For example, when I was describing feminine characters, often more of the stories that I told were more closed off. They were covered. The gestures that I used or the postures that I used were more again, inward facing, versus when I told stories of masculinity, often there were stories of conquering or of pursuing. And the way that I was told to like hold my body and carry my body was like more big and domineering of the stage. And this I found can lead to many like complicated and problematic gestures down the line, and had strong implications too for how I was conceiving some of these stories that I was portraying. So with this in mind, I both in my synthesis project did an analysis about previous dance pieces that I had done and that are commonly done within Kuchipudi dance narratives. And then I also worked on sort of analyzing how to break down some of these barriers and to use the art form to tell more positive stories, or stories that are a little bit more complex and are often underlooked in traditionally told stories. So with this, the first thing that I wanted to do is I wanted to stay true to sort of the roots of the art form. So I wanted to go through the process of creating the narrative, storyboarding, using that technique that I had been trained on for the last 15 years. And to do so, I took my first piece that I decided to do and sort of did a little step up piece in which I took a narrative that is commonly told, which is about a warrior named Karna. And I actually told a little bit of a scene of a story of his mother, Kunti. And usually the story of Kunti is a one-liner in a big book or a big story that is told. But I wanted to dig apart some of the story of Kunti a little bit more to give her character and sort of her experience more depth. And to do that, one thing that is often very quickly overlooked in a lot of Hindu narratives and Hindu stories is the fact that Kunti gave up her baby Karna very early on because she was a single mother and she didn't want to bear the social pressures of raising a child out of wedlock. So to actually be able to tell a story about Kunti's narrative, we wanted to stick to the roots by going back to some Sanskrit texts and finding a slokam or prayer that started to tell the story of Karna that the audience would standardly relate to. And then what we sought to do is, since we knew the message that we were portraying is one that is less commonly told, we wanted to use ragams, which are a series of notes that any trained Kuchipudi dancer or classical musician would understand and be able to recognize and immediately connect to an emotion. So we wanted this to help sort of the entire portrayal that we were telling through our story by portraying these ragams through the music that we used alongside the dance form to convey like these senses of sorrow and anguish and to give complexity again to the character that is often very quickly overlooked. Similarly, we had to go through the process of composing the music and creating a rhythmic sort of pattern to accompany the storyline that we were trying to tell. And we wanted to convey a sense of emotional intensity that often is not told in the same way that you would maybe describe like a battle scene or a conquesting scene. We wanted the scene of like giving your baby away and going through the pains of that to be just as rich and as deep of a story. So I would like to show you a little bit of what this would look like. It's a really short clip, but you can kind of see the different parts that come together in a dance work like this. So as you can see, that's a really short clip, but it kind of shows you the different elements that go together, everything from the costume design, and I know it's a little bit blurry through Zoom, but everything from the music, the costume design, to the specific motions that the dancers were using or the emotions that they were describing on their face were all kind of very carefully intentioned and designed to sort of add complexity to, again, something that is quite frequently overlooked. And we got a really positive response from this kind of work and this kind of approach. So something that we wanted to try to do was to actually take it to express more complicated narratives that are more closely tied to health. So what we sought to do next was to tell an LGBTQ coming out story. And the thing that we wanted to really highlight in this story was a mother-daughter relationship. And we wanted to be able to present this across a variety of different venues. So we presented this at Google's Diwali event, for example, and Stanford's South Indian Showcase. And we got surprisingly very emotional responses from the audience. And something that we found worked really well was using sort of the traditional hand gestures and sort of emotions that people can relate to. And in this case, that often was that mother-daughter relationship, and connecting it to a story that could be potentially harder to understand. And we had several audience members come to us on many occasions completely in tears, just saying like they finally found a sense of connection with the story and an identity that they never could fathom in their sense of the world before. And that was really powerful to us and sort of what we had hoped to elicit from this journey moving forward. So from the success of these two different sort of pieces and works, this led us to create an entire presentation called Ankaha, which we actually showcased in 2019. And it was kind of like my first starting point into sort of taking this way of modifying a traditional Kuchipudi art form to sort of tell these modern narratives. And in this showcase, we presented narratives on gender identity through Hindu literature using the same kind of process that I walked us through earlier. We used spoken word poetry to portray depression and to tell stories of mental health. And that helped us actually use poetry in English alongside sort of the hand gestures of Kuchipudi dance to target a slightly different audience. And we found that to be very powerful as well. And lastly, we also composed a story on child marriage, which was really powerful and went on to be presented in the UN. And this story actually is very dear to my heart because it was able to show sort of child marriage in terms of a personal narrative of a girl who is about to go and be wed off to her incoming husband. And I think it really helped kind of embody sort of a story and a narrative that is often viewed as a statistic or an us versus them problem. And that to me is kind of the power of arts and the power of using dance to tell these kind of messages to work on sort of cultural understanding. And this is only really just a start for me. You can see several other works that I have on this website, and I have the link. It's called tinyurl.ankaha, and I'll take you directly to the page where you can learn more about some of the works or the analysis of Kuchipudi dance. And Ankaha in and of itself means undercovered or untold. So we thought it was especially fitting for sort of this project. And yeah, this project has meant so much to me, and I'm super grateful for all of my mentors and for Dr. Sarnquist for kind of helping me work through this process. But I would love to get any feedback for any of you or ask for any questions that you may have. Thank you.
That's fantastic. Thank you so much. Thank you. I'm sure there are going to be questions. And I've asked you a lot over the years, so I'm going to defer. Lianne.
I have a question. Thank you. Shravya, amazing. I was wondering about how this has been received, right, by different groups of people, and if you could tell us a little bit more about that. Like, it sounds incredibly powerful.
Yeah, I think the reception and really understanding the audience as we perform these kind of pieces too has been very important for us, and choosing what we portray and how we portray it. Like I told you, some of the most positive responses have been people who literally came to us or wrote us stories who said, like, this has completely changed my understanding of other people and how I connect to other people, which is incredibly powerful. On the other end, we have gotten some criticism, but what we try to do really strongly is, even with these deeply cultural messages or religious messages, we try to ground it in less of sort of like, we try to stick as much to the books and as much to what is true to what people believe in, to work around culture rather than just causing more barriers, which has been something that was really important to us. So when showing these works, before we showed anything, we made sure to show it to different Hindu priests. We made sure to show it to different scholars. And some of the work moving forward, we're actually taking directly from people's narratives and seeing how they interpret their own experience. And that has been sort of a way that we've found to just say, like, we are conveying other things that people have told us is okay to convey. And that we thought was really important because we didn't want to distance more people or cause more boundaries than already exist.
Totally. Great, thank you.
Yeah, thank you.
That's really amazing. Okay, so I think we are going to go to a 5-minute break now. So we'll reconvene at 2:10 with a new set of students. But let's just give everybody in this session a giant round of applause. You all did great jobs. Thank you so much.
Yeah. Okay.
Okay, everybody, welcome back. For those who have just joined us, I just want to let you know that at the end of each of the presentations, there will be an opportunity for Q&A. And if you've got your participants panel up on the right side, you should see that you have the ability to raise your hand, and I will call on you and unmute you at that time. So welcome back to the second session. And with that, we're just going to go straight to Cameron.
Perfect. Thank you so much, Dr. Preston. I'm just going to share my screen really quickly. Okay, perfect. Hi, everyone. My name is Cameron Nosrat. Thank you so much for joining me today. So for my honors thesis, I decided to study the impact of recent climate extremes on mosquito-borne disease transmission in Kenya. So just a quick and pretty standard overview for today. I'll begin by giving some background on the subject and the research that's already been done, and then I'll dive into my study, specifically discussing the methods, results, and implications. So first, starting with some background. Climate change. It can have very important implications for human health. Climate change is projected to result in more intense and frequent extreme climate events, and these include droughts, floods, cold waves, and heat waves. This is important because both water availability and habitat temperatures have significant effects on mosquito abundance and growth, and ultimately disease transmission. Mosquitoes breed in pools of water, and temperature can affect many of their biological processes. Dengue fever is a mosquito-borne disease, which we will be concentrating on, and it's currently the fastest spreading mosquito-borne disease in the world, with over 400 million infections every year. So what research has already been done? Well, the effect of extreme rainfall on vector-borne disease dynamics is not well understood. Don't concentrate too much on the details of this slide, but just know that for increased rainfall or floods, there have been observed increases and decreases in vector abundance and disease transmission. And the same can be said for periods of drought. Temperature variability can also significantly influence disease transmission. It can affect the growth and maturation of mosquitoes and affect their biting rates as well. Our recent study found that each mosquito vector species has a different optimal temperature at which it best transmits various viruses. However, optimal temperatures for growth have differed between several studies. Overall, rainfall and temperature can have important implications on mosquito abundance and the transmission of vector-borne diseases. The purpose of this study is to elucidate how periods of extreme climate affect the abundance of Aedes aegypti mosquitoes and the transmission of dengue fever. So here are our 4 study sites in Kenya. We have 2 sites on the coast, Masambweni and Ukunda, and 2 sites in the west, Chilimbo and Kisumu. So now on to the methods. I wanted to compare the average vector abundance 1 month following an extreme climate event to the average vector abundance 1 month following a normal climate event. Then 2 months later, I wanted to do the same thing, but compare confirmed dengue cases. And this is the lag period that would be expected based on the typical mosquito cycle of infection. So for this project, I needed climate data, vector data, and disease data in Kenya. So beginning with the climate data, I extracted rainfall and land surface temperature data from satellite-based images. Then I calculated monthly averages for rainfall and land surface temperature. And then beginning in 2013, I took the difference between the observed monthly rainfall or temperature and the long-term mean for that month, giving me the anomaly. Then I classified these anomalies as either a drought, flood, cold wave, heat wave, or normal, based on whether it was in the upper or lower 10% of the anomalies. Then for the vectors, we were primarily concerned with this little guy, the Aedes aegypti mosquito. They prefer human blood meals and tend to live and feed inside of human homes. It's also the primary vector for dengue virus. So we targeted these mosquitoes at their different life stages using various trapping methods. We used an ovitrap to target Aedes aegypti eggs, pupal trapping to target them in their pupal stages, and then used Procopax and BG traps to record adult mosquito counts. And then cases of dengue infection were defined as children presenting with febrile disease who had a positive PCR and or IgG positive ELISA tested in Kenya and at Stanford. So now on to the results. So our first finding is that floods were observed to result in significantly increased Aedes aegypti egg abundance 1 month later. Here we have a box plot comparing average vector egg abundance 1 month later between normal rainfall, droughts, and floods. Egg abundance more than doubled 1 month following floods. We also conducted stratified analyses considering geographic region, urbanization, and individual study sites and found differences in these trends. So we decided to conduct a multinomial logistic regression as well. So this regression is adjusted for site, month, and year because we would like to account for between site and time endogenous variation. But as we can see, floods significantly increase the odds of having higher egg abundance. And when I say significant, like look at the red, I mean our results suggest a 1,280% increase in the odds of higher Aedes aegypti abundance following floods using a 1-month lag. For adult mosquitoes, which were recorded using BG traps and Procopacks, we found that cold waves actually increased the abundance of mosquitoes 1 month later. And this is a very interesting and novel finding because you would expect that as temperature increases, mosquito abundance would increase as well, since their biological processes and growth are sped up. And this is traditionally what has been found in the literature. So when we did our multinomial logistic regression to once again adjust for site, month, and year, we find that cold waves actually do not significantly increase adult abundance, even though we do observe meaningful odds ratios. It's important to note that year-round, Kenya has very hot temperatures, so cold waves were not really cold in that traditional sense, and they were actually more around 85 degrees Fahrenheit. It's also interesting to note that floods increase the odds of having higher egg abundance for adults recorded by BG trap, which is similar to what we saw with the Aedes aegypti eggs. In terms of actual cases of dengue, we conducted a similar binomial logistic regression adjusted for site, month, and year to observe how extreme climate events affect the risk of dengue after 2 months. And interestingly, we do see heightened risk during floods by about 146% and 164% after cold waves. Even though these aren't significant, they are meaningful and they are expected based on our previous mosquito abundance results. So looking forward, what do these results mean? Extreme climate events increase the risk of mosquito-borne disease transmission, and more specifically, floods significantly increase the abundance of mosquitoes and the risk of dengue transmission. Floods seem to have provided additional breeding sites for our mosquitoes, allowing for greater transmission. So moving forward, what can we do about this? Well, in Kenya and around the world, breeding sites can be artificial and natural. What we can do is do our part to get rid of these artificial breeding sites, and that begins with trash. Trash like tires, garden pots, etc., when left outdoors can provide ideal breeding sites. So being sure to remove these can limit the reproduction of mosquitoes. In order to prevent transmission as well, it's important to be cognizant that Aedes aegypti mosquitoes primarily live and feed inside of homes during the day. So bed nets, which would work with other species, will not necessarily work with Aedes aegypti, and they can be a little more difficult to target. Using indoor spray and repellent can be helpful as well, but overall, further studies with higher temporal and spatial resolution are necessary in developing models to predict and inform people of when they might be at an increased risk for infection following extreme climate. Thank you all so much for your attention, and a special thank you to everyone that helped me in conducting this project.
That's great. And we already have a question from Zach.
Hi. First of all, great job, Cameron. That was pretty interesting, and I didn't know pretty much any of that. My question for you is, throughout this process of whether it was when you were compiling information, running your analysis, or going through conclusions, was there anything that you found particularly noticeable or contrary to what you thought you would find about the data or the conclusions, given either like your prior knowledge about the subject or just like if there was anything you could comment on from that perspective that you would think would be worth noting?
Yeah, that's a great question. So I guess the most surprising thing, as I mentioned, was that cold waves were actually found to increase the abundance of Aedes aegypti adults. And that hasn't traditionally been the case because you would expect that with higher temperatures, mosquitoes are able to grow more and there would be a greater abundance. I think specifically our results were driven by the fact that these cold waves weren't actually cold. They were cold in the sense that they were an anomalous period of cold temperature, but they were under periods which would be defined as pretty warm weather. So I think that those warm temperatures during the cold waves, if that makes sense, was driving the results that we found there.
All right, Lianne.
Oh, great presentation, Cameron. Super interesting. I was wondering from a modeling perspective, why you chose to dichotomize some of your outcomes Yeah, why that choice? Often you can lose a lot of power when you do that.
That's a really good question. So the main thing was that all of our outcomes, whether that was vector abundance and dengue cases, were non-normally distributed. So we required the use of non-parametric tests, and more so, there were plenty of outliers on the upper end. So we thought that that would skew the results significantly. So that's why we decided to categorize the variables in that way.
Nice, excellent answer. Thank you.
Great. Yeah, that is super interesting and relevant research. Thanks so much, Cameron.
Thank you.
Okay, we are going to move on now to Sierra, who is another one of our synthesis students, and we're going to kind of continue on the theme of disaster. So I'll just hand it over to her.
Sounds good. Let me get my slides up here. Okay, can y'all see those okay? Hopefully.
Yes, it's good.
Cool, sounds good. All right, so I started out a year ago thinking that I was going to be researching just like the general possibility of a pandemic happening in the distant future and looking at some of the problems that might lead to like a pandemic becoming even more of a problem. Over the course of the past year, that's unfortunately transformed into something much more specific and has become more focused on the coronavirus epidemic, as well as some of the factors which have contributed to this epidemic becoming so big. In this presentation, I'm going to talk a fair amount about the process of coming up with this project and going through an ongoing pandemic while trying to write about that pandemic and all of the problems with that, as well as the final product that I've come out with in the form of a couple of different articles about this subject. So first, let's talk about the process. I started out in the fall when I began writing and researching this by looking at a report which came out in September of 2019 by an organization called the Global Preparedness Monitoring Board. This report, A World at Risk, was intended to essentially provide a warning to the world and to policymakers that the world was not prepared for a pandemic or health emergency. The hard part with this is that it wasn't particularly accessible or effective at communicating information to non-policymakers, just like your general lay audiences, the people who to some extent really do need to know about this so that we all can put pressure on policymakers to put attention to this beyond a single news cycle. So in my project, I wanted to turn to the sort of themes of accessibility, effectiveness, and accuracy to make sure that I could convey something about the contents of that report and some of the general factors that went into it for lay audiences, for people who might not necessarily be experts or need to know the policy analysis, but want to know what's going on in the world in general. I started out in the fall trying to write a warning. I thought, okay, well, there might be a pandemic in the distant future. We all should be very warned about this and know that it's a problem so that we can prepare. As time went on, I realized that some of the issues that I was researching and the things that seemed like the biggest problems were the fact that there is a lot of anti-science sentiment right now. There are a lot of people who are against vaccination and a lot of people who feel like maybe we don't necessarily need to put so much trust in science as a whole. There's also a lot of distrust of authority, which seems to be one of the big risk factors, or so I thought at the beginning of my research. And I also thought that there was just like general incompetence, which the report goes into a lot of detail on in terms of lack of coordination and just general lack of readiness. In October, my conclusion was pretty simple. One of the things that I wrote sort of ended in the idea that this report might simply not be heard, even though it had already been written. As time went on, though, I sort of began to enter this phase of impending doom or panic, realizing in January, as we had a coronavirus epidemic that was starting to make its way around the world, that this is something that's going to happen soon. And so I began to write more in detail about the coronavirus outbreak. In January, I wrote, "How does society cope with uncertainty in an era where pseudo-certainty abounds? This coronavirus outbreak suggests a very simple answer: not well." Unfortunately, that's become more and more clear as time has gone on, as we see that there's differences between governments that claim they know what they're doing and governments that don't even pretend to know what they're doing. And as time went on, I also started to become more concerned about the way that these tensions would present themselves. In March, I wrote that I expected within the month that warnings of contagion would turn to calls for self-isolation in the United States if the White House didn't prevent them. So over time, I've kind of started with this like broader research and slowly started to narrow my way down, throwing out my chapters from the fall in favor of more in-depth chapters and research, and eventually towards form of articles. So by the time we reached March through May, I focused more on telling the story of this pandemic in the context of past pandemics and in the context of some of these broader tensions that I've talked about throughout this. So I wanted to focus then on the idea of trying to portray disaster in a way that was clear and understandable. Now I'll turn to the product, the articles that I've written. For this, I'll go through each of the articles very briefly with a summary and then read a little bit of an excerpt from two of them. The first article that I wrote was written literally the night that I packed up my dorm room to leave Stanford for good. It was called "It's on Us," and this article was intended to convince people that they should take individual action more seriously as possibly the only strategy we could use given government inadequacies. So this article was directed at people in my general vicinity and published on a blog called covid.blog or Health Attacked, which I created to post these articles. My second article focuses on that report, A World at Risk, and talks about the ways in which our engagement with that report and with general attempts to call the warnings on pandemics for decades now have been delayed and denied. Instead, the world decided to risk it. My third article looks at a comparison of the response to SARS and the response to the coronavirus, specifically narrowing in on New York State in terms of both the lack of coordination between the state, federal, and local governments for New York City, and on a general attempt to cover up or pretend that the virus was not as good or not as bad as it actually was. This article, passed as prologue, tries to sort of track those different trends in terms of how response is carried out. The next article focuses on the long term and some of the uncertainties that we have in terms of how we're going to deal with this coronavirus. My second last article then is called "The Shaky Future, COVID-19 in the Long Term," and tries to analyze some of these tensions that we've seen throughout, some of the tensions that we can see from past pandemics, and the things that we can expect to see in the long term as far as I can guess or as far as anyone can guess. The last article takes a little bit of a turn towards the thing that I actually concentrated my entire HumBio major on, a concentration that I made up called disaster public health. And in this, I tried to look at the case for disaster public health as a mechanism of combining disaster management strategies and a focus on resilience and recovery, which we can find through public health mechanisms and strategies. So from all of these then, I essentially am trying to analyze the story or tell the story of this virus in a way that's relatively understandable. I want to turn now to a brief excerpt from my first article and a brief excerpt from the long-term article. One of the ways that I like to think about the pandemic and the pandemic response is through the lens of whitewater rafting. So my excerpt is as follows. As someone who grew up whitewater rafting every summer, I like to compare communal disasters like this and the wildfires which destroyed part of my hometown to entering an intense rapid on the river. To make it through, especially when conditions are changing quickly, you need to buy time for the guide, sitting at the back to assess the situation. You fight with the current to dig your paddle in, alongside the rest of people in the boat, working to keep the boat in an eddy above the rapid. You back paddle with every ounce of energy in your body, knowing that every second of time you save for preparation increases the odds that you'll make it through okay. If you don't trust the guide and think they need more time, you work even harder. You back paddle while your arms hurt and your individual effort doesn't feel like enough. Feels like simultaneously the greatest sacrifice and the greatest failure. But if you all work together, you can make it through. At the time, I wrote, right now we're sitting in that raft above the rapid, and it's time to dig in. It's on all of us working together to hold back the boat before the onslaught, because if we don't buy our underprepared local health departments and hospitals time to guide us through this, many more thousands or even millions of people will die. I want to end with an excerpt from my long-term article. But right now, as things begin to open up around the country and the world, we're no longer waiting at the top of the rapid to dive in. Instead, we are working in the middle of the river. Around every turn of this river of hellish experience is another rock, another potentially devastating outbreak that could pop up if we don't maneuver carefully around it. If we get stuck on one, the boat might flip, losing us entirely. But if we're careful and use a balanced approach, we might be able to make it through this without the massive surges in hospitalizations and deaths that we all must fear. Obviously, a lot of people haven't had that chance, but I hope that by writing these articles and by trying to combine these different strategies and look at some of the connections between disasters and public health, I'm hopefully able to give a different insight into all of this and make it a little bit more accessible for all.
Fantastic. I think we have time for one question. So whoever is the fastest hand up. Oh, see, now I've discouraged people. Okay, so, Lianne.
Mute myself. Fantastic, Sierra. So I love that they're on your blog. Where else might these pieces go to sort of promote their exposure? Do you have ideas for where you could send them?
Yeah, so when I initially set out to write this, I thought that my articles were going to be like, I don't know how to describe it exactly, I guess like a little bit more lighthearted. I think now it's a little bit less lighthearted, but I'm an editor for Stanford Politics, so I've considered trying to maybe publish one or two of them there, or just try to make them more visible and clear on my blog, because I plan to continue it since it's kind of dedicated to disaster public health in general.
That's great. Thank you. Thank you. And I just have to say, I love this idea of pseudo certainty, because that's kind of dangerous.
It's a big problem.
It's a problem. Okay, we are just going to move right on to our next speaker, who is Max Klotz. Max, are you ready?
Yes. Can you hear me okay?
Yeah.
All right, let me just share my screen. Does that look good? All right. Hello, everyone. My name is Maxwell Klotz, and I'm going to start today with two paradoxes necessary for understanding national parks. The first is that national parks exist both for visitation and for preservation, two goals which are often at odds with each other. And the second is that people think parks are pristine, unchanged wilderness, but in reality, they've been shaped for generations by human influence. What's important with these paradoxes is that visitors have a perception or imagination of iconic national parks before even arriving. And this dynamic has shaped both Yellowstone and Zion. In this project, I explored these paradoxes and others to investigate the relationship between the cultural imaginations and scientific realities of both Yellowstone and Zion. I used a combination of archival materials I obtained on site in both parks with secondary research materials, including historical articles and books and scientific ecology articles. And I thought that this interdisciplinary approach would provide the proper historical information necessary for contextualizing environmental changes over time. I selected Yellowstone and Zion for this study for a number of reasons. First, their cultural prominence. In 2019, they were the 4th and 6th most visited parks in the United States, Yellowstone being 6th, and Yellowstone was also the first national park ever established in the United States. Beyond their prominence, they have a number of contrasting elements that lend for a rich comparison, including their differing ecosystems, different sizes, with Yellowstone taking up parts of 3 different states, Montana, Wyoming, and Idaho, whereas Zion only occupies a corner of southwestern Utah, and different experiences on the ground in each park. And traveling to the parks helped clarify these different experiences for me. And when you're in Zion and Yellowstone, you realize that both parks are creatures of how you get around. In Zion, this is on the shuttle, in Yellowstone, in personal vehicles. And there's no understanding how the parks evolve without understanding the transformation of transportation across the history of the parks. It started with railroads, which had a heavy hand in the development of both Yellowstone and Zion. They brought visitors in, they advertised the parks, and created guidebooks on how to visit. And this sets up a paradox of commercial involvement, where in the founding documents of both Yellowstone and Zion, they are set aside from commercial exploitation. However, almost immediately, commercial enterprises become involved in park operations. And both the Northern Pacific and Union Pacific Railroad companies sponsored artists in the early days of park discovery to paint and create visual imagery that would draw up support for both founding and visitation. This is one of the more famous such images painted by Thomas Moran, the Grand Canyon of the Yellowstone, and many historians cite this as key in getting the park founded in 1872. Over the course of the 20th century, transportation shifted from railroads to automobiles, and in another paradox, human-made automobiles became integral to visitors' experience of wilderness. Now, cars brought people closer to animals and to park spaces and made it easier than ever to get to the national parks, and this coincided with a rapid increase in visitation. If you look at visitation statistics across the 20th century in Yellowstone, there's a steady increase throughout the 20th century coinciding with the spread of automobiles and highway construction. And now the park receives just over 4 million visitors per year. In Zion, there's a similar climb throughout the 20th century, albeit slower, coinciding with automobiles. And recently, starting in 2013, another spike correlating with the rise of Instagram and other social media platforms. And these platforms are almost acting as modern day Thomas Moran's, drawing in visitors with photographs and artwork. Now Zion receives 4.3 million visitors per year. And interestingly, in both parks, visitors are still seeing many of the same signature sites as were being advertised in the early days of the parks, such as Old Faithful and the Yellowstone Falls. Now, visitation came with a number of ecological impacts. In the early days of Yellowstone, park policy was geared towards creating a paradise for game animals, which meant removing predators, leading to the complete elimination of animals like wolves from the park in the 1920s, and they did not return until reintroduction in the 90s. And tourists also brought in invasive species, which threatened the biodiversity of native species, such as the cutthroat trout shown here, which is threatened by invasive rainbow trout. In Zion, the main ecological impact, while those two are still present, is crowding. Crowding affects popular trails with increasing erosion, pollution, and just negatively affecting park experience when you feel like you can't get away from visitors. Now, these parks are sure to continue facing these threats in the future, but there are also going to be new threats. Climate change, for instance, will force or allow animals to move into higher elevations, disrupting existing habitats. For instance, the mountain pine beetle, which when it moves to higher elevations can disrupt whitebark pine forests. Secondly, crowding is continuing to increase in both of these parks, and they may reach their carrying capacity in terms of visitation. Zion is much closer to this, but we run the risk of loving these parks to death through our overzealous tourism. And the third is development outside the parks, which can lead to habitat fragmentation as animals rely on migration corridors for winter ranges. And these are threats that not only these parks face, but other national parks and wilderness areas around the United States as well. And it will be key to understand these threats in order to manage these parks going forward. So I'd like to close with another paradox, that national parks exist for the benefit and enjoyment of the people, but also have a mission to protect their land in perpetuity. And at odds, or at times, the present desires of tourists and visitors can impair the ability for future generations of the park to enjoy it in the same way. So it's vital to understand that national parks simultaneously exist in the past, present, and future. Their park management is largely geared towards recreating an ecosystem which hasn't existed since Europeans arrived in that area of North America. In the present, they are geared towards drawing in tourists, and in the future, they're geared towards preserving this environment for future generations. So in order to properly evaluate park management going forward, we have to understand that the parks exist in these 3 timeframes simultaneously. I'd like to extend my sincere gratitude to my 3 readers, Richard White, Catherine Preston, and Peter Vitusik, who provided me with invaluable guidance along this whole journey starting back in fall 2018. I'd also like to thank Undergraduate Advising and Research and the Bill Lane Center for providing me the grants necessary to do archival research in both of these parks. And finally, my family and my friends, particularly my mom, for helping me as a research assistant at times and as an editor. And without all these people, my project would not have been possible. So with that, I will open it to questions. Thank you.
We have a couple of questions, but I'm going to actually start with one that was put into the chat. So Patrick Shea says, there's been some effort to require a reservation to visit some national parks. Should this be implemented in the most popular national parks?
Yes, so this measure was first proposed in Zion, I believe, because Zion, while being one of the most popular parks in America, due to its geologic structure of being a small slot canyon, is uniquely affected by crowding. So in talking to the current chief of commercial services in Zion, it sounds like the reservation system is sort of in their consciousness, but it's more of a future plan, maybe 10 years down the road or 5 or 10 years down the road, as opposed to the near future. It seems like they're trying to implement smaller measures in the meantime, like day use permits for their most popular hikes. I think that certainly in the next couple decades, we will get to a reservation system for parks like Zion. I think in Yellowstone, that's farther off because Yellowstone is so huge and vast. It allows visitors to spread out more evenly. But I think of a reservation system, particularly in Zion, if not in the near future, will be implemented at some point.
Okay, and Annette has a question.
Hi, I really enjoyed seeing the pictures of nature. That was a fun part of the presentation. It was related to the first question, but maybe going a little bit more broadly. In addition to reservations, a reservation system, do you have other ideas that might be ways that we could improve park management to prevent some of these threats to the parks?
Yeah, so on the crowding side, there's the reservations, the day use permits, perhaps extending something like the shuttle system into other national parks, maybe on busy roads in Yellowstone, like the road that connects the west entrance to the Old Faithful Geyser Basin. But aside from crowding, with development particularly, because these parks, while we think of them as being sort of controlled wilderness areas, their ecosystems extend beyond the boundaries of the parks. So Yellowstone is part of the greater Yellowstone ecosystem, and animals migrate outside of the park to lower elevations during winter months because it's a more temperate climate. So some measures that have been proposed to allow these animals to migrate outside include creating migration corridors across highways in Wyoming and Montana that would allow animals to cross on their normal migration routes, which have been disrupted. Also partnering with private landowners to allow animals to refuge on their land during winter months. As far as climate change, the impacts of that are still fairly unknown. We know that species will move and ecosystems will change, but it's not clear exactly what the extent of that will be. So that is more of a wait and see approach.
Okay, I'm going to go real quick to Conrad for the next question. Conrad, I tried to unmute you, so don't, okay.
Hey, Max, I thought you did a fantastic job. I wanted to ask a question a little bit about the trajectory that you mentioned in terms of increasing visitors at the parks. I was curious if you could speak about some of the factors that you think are contributing to that growth in visitation, whether we can expect those trends to continue, and then, I guess whether you see that transition to more visitors is a good thing for the national parks broadly, or whether the crowding kind of outweighs any potential benefits of getting more people to see the parks.
Yeah, so like a lot of things in this project, it goes both ways. It's paradoxical. Visitors coming into the parks raises funding for the parks and contributes to revenue, which is great, but also, as I've showed, they have a number of impacts which can affect both the park ecosystems and just the park experience for other visitors. It seems like the big spikes in visitation have happened, partially because of social media, particularly in Zion. It went from 3 million visitors in 2010 to this past year, it got 4.6 million visitors. So social media is a big one. Yellowstone has been more of a steady increase, which has leveled off somewhat recently. So I don't foresee any real spikes coming with that. Everyone kind of knows what Yellowstone is and knows what to expect going there, or at least has an idea of what to expect going there. And then some other factors increasing visitation are international tourists, particularly there's a large fascination with the American West among both Europeans and Eastern Asian tourists. So if you go to national parks now, there's a huge proportion, I don't know the exact numbers, but a huge proportion of international tourists. And this has contributed a lot to the growth in visitation.
Okay, fantastic. I'm going to move on now to another synthesis project, the last person in this session, Julia. And
thank you for joining me for the discussion of my human biology synthesis, which is a study of the crossover of arts and medicine historically and in the modern era. My name is Julia Todderud, and I am grateful for the support of my advisor, Lisa Medoff, on this project. Through my synthesis, my hope has always been to take an integrated approach to address the crossover of art and anatomy through a historical analysis, a modern look at the divide between the two, and an art series to put it into practice. While historically there has been a connection between arts and medicine, the modern scientific community separates and stigmatizes this dynamic. Beginning with a historical approach, early study of anatomy was developed and spread through art. Paintings, drawings, and prints became our way of communicating the different aspects of the human body. To address this historical example, my intent is to use a case study of two key artists, those being Leonardo da Vinci and Andrea Vesali, who are key in revolutionizing our understanding of the human body. Beginning with Andrea Vesali, he wrote a key text, that being De Humane Corporis Fabrica, which translates to On the Fabric of the Human Body. He had spent many years studying at the universities of Paris and Padua, and he would sneak into graveyards to steal corpses and perform dissections. But what he ended up finding was that the lessons his teachers had provided were discordant with what he observed in those bodies. He used this information to create what later would become a template for the human body in a text that is often considered the foundation of modern anatomical studies. Through his images, he portrayed musculature and different systems of the human body beautifully with a long-lasting impact. Often more familiar is Leonardo da Vinci. To the right, you can see his figure, the Vitruvian Man, which portrays the proportions and ratios of the human body. But he also created an extensive series of anatomical sketches, which you can see before you. I had the fortunate experience of being able to handle these drawings personally, and they are just absolutely incredible up close. And surprisingly, his drawings were incredibly accurate and in accordance with anatomical structures seen today. Both Da Vinci and Vesalius were revolutionary in their time and continue to inform and intrigue medicine in this modern era. Through their artwork, we were able to learn about incredible structures of the human body that we weren't before familiar with, as prior to their works were released, much of what we understood about the human body was based off of dissections of animals. And human anatomy and animal anatomy are not the same. Key figures we learned about were cardiovascular systems, musculature, the reproductive systems of both men and women, and connection and motion, that being in joints and tendons and how the body functions as a whole. In modern times, there's a very prominent divide between the arts and the sciences. Arts are associated with creativity and humanism and just pure imagination, while sciences are more rational and definitive with a lot of reality testing to make sure that everything is very accurate to the experience. Now, both of these have incredible benefits and both perspectives are very strong, but this divide has resulted in a lot of detriments to our communities. One author described the divide between the two as such: we look to science to find solutions for practical problems in the hope for a better world, and to artists to borrow a personal vision of the experience of life. But by having this divide between the two, we see a lot of different barriers come up. In my own college experience, I've seen this extensively, that being STEM fields versus humanities, techies versus fuzzies. It's a very academic divide. There's also the issue of reputation. Artists and scientists have a very, very different reputation, which is the reality, but very unfortunate for both fields. There's also the focus in medicine on only that which is testable, which makes it very difficult to incorporate arts and have that creativity in there. As a result, we see this lost tradition. We see this loss of the connection of arts and medicine, and we in fact see the stigmatization of the arts within the medical field. This divide helps us to lose a great deal of benefits that we could have. In my own experience, when I took my first anatomy course, I hand drew over 500 flashcards featuring the different anatomical structures we were expected to memorize. In this application of art, I was better able to understand and learn how each aspect of the human body works together. We learn through visual acuity, the visual learning and hand-eye coordination that can be gained from the arts. There's the creativity associated with the arts that come into play in medicine through innovation and imagination, developing the very transferable skill of creative problem solving. There's also the core aspect of human connection, which is quite often very lost in the medical community today. But through the arts and through different aspects of the artistic communities, we're able to gain a whole person understanding, having a more wide perspective. We also improve our ability to connect with others and increase the ability to access those emotions and communicate those with others. Now, I briefly mentioned my experience before, but to extend my human biology synthesis, looking not only at the historical and modern challenges, I hoped to put this into practice to really improve those skills that I think that so many more people could benefit from. And I created my own anatomical art series. I began with several sketches in the styles of Da Vinci and Vesalius, focusing on modern conditions that weren't known about then, such as scoliosis on the left and Alzheimer's on the right. I also incorporated other sketches like heart disease and asthma, just really trying to push my understanding of the body and conditions that we know today. I started to push this a little bit more in my own painting practice to see what different aspects of the human body we can associate with identity. I called in Andrea Vesali's figure on the left into my own painting on the right and found that it really helped me to understand the skeleton of the human body. But I also just found the results of this to be really beautiful and a really wonderful practice for me. I pushed it a little bit further to try to see this in my own practice and pulled all of Vesalius's figures into this art series, which I quite enjoyed making. Overall, I found that these studies of the human body really allowed me to experience anatomy, but also to connect with this historical connection and see how we could connect it to modern medicine. So thank you all for your time, and please let me know if you have any questions.
Fantastic. That was amazing. Okay. Questions for Julia? Samantha.
Hi, Julia. It's so awesome to like finally get to see your artwork. I've been kind of looking forward to that for a while now. It looks like you used a few different media to make those. So was there one that you preferred over the others, or one that like helped kind of make it feel more anatomically correct?
Absolutely. Thank you for your question. I think that, I mean, painting is my preferred medium for most of my works, just because I really enjoy like the texture of it and really working with it. And I think that in the structures that I portrayed of the body, it allowed for a lot of like fluidity. But I also really enjoyed working with like ink and pen, because I think that also had a really beautiful gesture to it that really called in that tradition of like Da Vinci and Vesalius, and really allowed me to kind of have that expression of the human body rather than just the basic structures.
Yeah. There's a question in the chat too from Patrick Shea, who says, how does the world of digital art fit in your effort to bridge science and art? And are you selling any of your paintings?
Well, I can I can absolutely sell my paintings. Some of them are currently locked in the dorm rooms on campus, so a little bit harder to access. But I think that digital art is obviously, it's just as valid as any other form of artwork. And in fact, it's really interesting to see how the portrayals of the digital human body vary from the more gestural painting and drawing depictions. So I've really enjoyed seeing that, because a lot of what we end up seeing in the digital artwork is very, very anatomically correct, because you do have more capabilities to enter parameters. And also, if you look at textbook illustrations, or there's a lot of different websites where you can utilize artwork to display the human body in 3D forms, and you can almost virtually dissect the body. And that's definitely just as beautiful. It's just as educational. And while digital art isn't personally my style, I absolutely respect and recognize the efforts of the people who do participate in that.
Okay, great. And then one last question is Jennifer Wolf.
Julia, I have a quick question. Do you still have those 500 plus flashcards? And it would be fascinating to be able to analyze those as data to see the use of art in learning specifically, but do you still have them?
Yes, I do still have them. It's a huge stack of flashcards in a plastic Ziploc baggie. And my intent is at some point, when I come up with what I feel is a good enough idea, to incorporate them into an art piece themselves, because I think that would be a really kind of a full circle moment for me on that. I mean, as part of the literature review I did for my synthesis prior to writing the paper and prior to doing my art series, I did learn a lot about how that visual learning, how that art as a learning method is really, really effective, especially in courses like anatomy, where it's so important to understand like layering of different structures and the interaction of those different structures and those points of connection. So I definitely, in my own experience, it was very, very effective, and it's often a learning tool that I use. And I have seen how that it has really impacted others as well in the analysis of those learning methods.
Thank you.
Fantastic. Okay, so we have, let's give everybody in this session a round of applause. And we are going to just take about a 5-minute break and come back just a little bit before 3:05 to keep us on track. All right. I think we're mostly pretty much all back. Oh, I need to, sorry, start the recording again. So welcome back to this session. And just to let people know, if you haven't been here before, at the end of each of the presentations, there will be time for questions. And if you can reveal your participants panel on the right, you'll find a little blue raise your hand. So if you raise your hand, I'll call on you, I will unmute you, and then we can go from there. So let's just jump directly to our first speaker for today.
Great. All right. Hi, everyone. Can you see my screen? Okay. Okay, great. So, hi, everyone. My name is Heman, and today I'm going to be presenting on my honors thesis project, which is titled Understanding the Immune Response to Malaria, Assessing the Relationship Between Antibody-Dependent Cellular Cytotoxicity, ADCC, and Innate Cell Phenotypes in Children from Tororo, Uganda. And so just to give a little background on malaria, it's a disease that affects around 228 million people around the world, resulting in around 405 deaths each year. And a majority of malaria-related deaths are due to infection with the Plasmodium falciparum parasite, which is one of many parasites that are responsible for infection. And a majority of cases occur within sub-Saharan Africa. And interesting research has shown that children living in malaria endemic regions can eventually develop what's known as clinical immunity to malaria. And clinical immunity to malaria is a protective mechanism that has been thought to be responsible in the partial control of the malaria parasite, as well as control of symptoms of the disease. And so in populations where there's repeated exposure and increasing age, it's actually been shown that there's less symptomatic infections and actually an increased probability of asymptomatic infections. And so if you look to the graph to the right, you can see that in infected children living in areas with a high incidence of malaria, shown by the red line, they actually develop lower parasite densities or lower parasitemias with increasing age. And the exact opposite is shown for children that live in lower incidence areas, shown by the green line. And so this forces us to ask a question: what are mechanisms that are driving this naturally acquired anti-malarial immunity? And cells that are positive for a specific marker known as CD16 can help to answer this question. And so research has shown that certain subsets of natural killer cells, which are immune cells that are positive for a marker known as CD16, has been correlated with decreased parasite densities. And so if you look to the graph to the left, you can see that with a higher percentage of CD16 positive, CD56 negative NK cells, there's actually an association with a reduced parasite density over time. And so this translates over to my project specifically because we were analyzing two subsets of NK cells, which were CD56 bright, CD16 negative NK cells, and CD56 dim, CD16 positive NK cells. And so you might be wondering, through what mechanisms do the cells that are positive for this cell marker known as CD16 lead to decreased parasite densities? And it's through the mechanism of antibody-dependent cellular cytotoxicity known as ADCC. And so this is a general schematic of how ADCC works. So first you have a target cell that is infected with malaria, and it'll present a malaria parasite antigen on its cell surface. And then you'll have antibodies come in and recognize this antigen, forming a complex. And then you'll have an effector immune cell come in, and in our case, it's an NK cell, and it'll recognize the FC region of an antibody with the help of this CD marker known as CD16 that we just talked about. And the binding and recognition of these two cells will then eventually lead to what's known as granule polarization, and then eventually cell death. And so for this project specifically, we were looking at, are there any correlations between ADCC performance and immune cell phenotypes of natural killer cells, as well as other immune cells like gamma delta T cells, monocytes, in 19 samples from children living in Tororo, Uganda. And Tororo, Uganda is a malaria endemic region within sub-Saharan Africa. And then we also looked at whether there's an association in sex and performance in ADCC. But for this presentation specifically, I'm going to be focusing on the results that we found specific to NK cells, and then any association between sex and ADCC performance. So, oops. Okay. So data was collected by conducting ADCC assays on 19 Ugandan participants. And so this involved one participant that was collected with their sample being collected in 2013, and then 18 of them being collected from 2017 to 2018. And these assays were then performed, and then data was collected through a lab technique called flow cytometry, which allows you to analyze physical and chemical characteristics of cell populations. And then after obtaining this data, detailing how well these cells from these samples are able to perform ADCC, we then compared them to cell phenotype data available from 2 different time points. One time point being from data that was available at 2019 that was paired with the time that we did the experiment, and then 2013 to see if there are any associations between cell phenotype and ADCC performance. And so one of the first interesting results that we found was that there was a higher ADCC rate that was reported in females compared to males. And so from the graph shown on the top left-hand corner, it's been consistently noted from our colleagues at UCSF that parasite prevalence is higher in males than in females. And if you look at the graph at the bottom left, duration of malaria infection is also longer in males than in females. And so looking at our data to the right, it shows that even though there's not a statistically significant correlation and that it's analyzed in a sample size of around 19 people, there is a trend that indicates that there's higher ADCC performance in females compared to males. And this is interesting because this can further help to understand some of the results that are shown on the left-hand side from our colleagues. And so as I mentioned before, we also compared ADCC performance to cell phenotype data that was available from 2019 as well as 2013. And this slide shows that one of the specific cell subsets that we were looking at, CD56 bright NK cells, could be only used for accurate comparators to ADCC because there isn't a statistically significant difference between the 2 data sets at the 2 different time points. But the same thing can't be said for CD56 DIM cells because there's a statistically significant variable difference between the data that's provided in 2019 and 2013. And so when looking further into the data, we're able to see that CD56 DIM NK cells didn't significantly associate with ADCC performance, while there was a statistically significant negative correlation with CD56 bright NK cells and their performance in ADCC. And this is interesting because it might be that these cells aren't performing ADCC because they aren't expressing the specific CD marker known as CD16, or that they might just be mediating cell death through some other mechanism like increased cytokine release, but we won't know until we conduct further experiments and until further research is conducted on that. And so just to summarize the results, ADCC was reported at a higher rate in females compared to males. There was no significant association between ADCC and CD56 DIM cells, and there was a statistically significant inverse correlation in ADCC associated with higher CD56 bright NK cells in the paired data that we had in 2019. And so some limitations during this project involved the small sample size that we were analyzing with 19 people. And then there were also frequent outliers within the data. And another limitation was that this project hinged on the fact that we were looking at retrospective data that was collected in 2013, when 2013 wasn't a strong comparator to use looking back at the results that we had. And so we also plan to use more than 19 samples, but given technical difficulties that I had in setting up the ADCC assay, as well as restrictions that were placed during the COVID-19 pandemic that prevented us to go into lab, this was the reality of the project. And so next steps would be to conduct ADCC assays with paired markers rather than relying on earlier data from 2013, and then to obtain a larger sample size in order to ensure the validity of some of the interesting results that we found from this project. And I just wanted to take the time to acknowledge and thank the following people for all of the help and support that they've provided during this project. Without them, I couldn't have completed this thesis project. Thank you so much.
That's great. Good job.
Thank you.
Questions for Heman? Yes, Lianne.
A great presentation, Heman.
Thank you.
You're welcome. What might underlie the sex difference that you observed?
What might underlie it? Yeah, so it could be a variability of different things, and because ADCC is one out of many mechanisms that are hypothesized to lead to this idea of clinical immunity. So it might be that females are performing ADCC through the immune cells that they have, because there are multiple immune cells that are implicated in this process, or it might be one of the many other reasons, like whether if there's differences in interferon gamma or like other cytokines that are responsible in this process. And so from that data that I showed, it's indicative that females are performing ADCC at a higher rate than males in their immune cells, but you have to conduct further assays and experiments in order to actually conclude that, because it was such a small sample size.
Okay, fantastic. Any other questions? If not, then we're going to move on to our next speaker, who's Laura Jonsson.
Hello, can everyone hear me?
Yeah.
Wonderful. Okay, let me share my screen. Okay. Hello, everyone. My name is Laura Jonsson, and for my honors thesis, I ran a pilot study exploring home language environments in rural China. So it is well understood that children's early language skills are important for their development and outcomes later in life. Now, what drives this early language development? It is primarily caregiver-child interaction, but we know that there is substantial variation in what this looks like across cultures. A recent study in rural China found that half of the children under 3 years old in rural areas had a language delay. Now, researchers hypothesize that this might be due to low rates of interactive parenting, such as reading and playing with the child. These are typical home environments for a child in a lower socioeconomic status or lower SES family in rural northwestern China. But how do we document the potential differences in language outcomes between children? We do so by measuring a child's home language environment, which was the first objective of my study. I collected data last summer with my wonderful colleagues shown here in China, and we traveled to 16 rural villages in 5 different counties across Shanxi province, located in northwestern China. Our participants were 38 young children who were roughly 25 months old, and all came from lower SES backgrounds. Only 21% of mothers and 11% of fathers had at least an upper middle school education. We utilized Lina recorders, which are placed in a specialized t-shirt that a child will wear all day long. The Lina software then automatically produces a count of all adult words near and clear to the child, and also a count of conversational terms, measured as the back and forth between a child and adult. Previous studies using this device in English and Spanish have shown wide variation in these measures, even within the same sample of children. So what did I find in my study? First, we found that our participants heard, on average, 15,000 adult words throughout the day of the recording. We found no significant differences in average adult word count between counties, meaning that no county was clustered at the top, middle, or bottom of this range. Finally, these results indicate striking variability for the whole sample, with a 5.5-fold difference between our lowest ranking child and our highest ranking child. Now, what about conversational turns? Our participants engaged in approximately 600 conversational turns throughout the day of the recording. Again, we found no significant differences in conversational turn count between counties. And we see a similar variation within the full sample, this time a 6.6-fold difference between our lowest ranking and highest ranking child. Now, how do these home language environments of my participants compare to existing data? To answer this question, I compared my sample to first Zhang et al.'s sample of urban higher SES families in Shanghai, China, and secondly to Weisleiter and Fernald's lower SES Spanish-speaking families in the United States. So let's see how my mean of 15,000 compares to that of Zhang's sample. Their sample heard roughly 28,000 words. Zhang also divided their sample into upper and lower halves based on adult word score. So to make a direct comparison, I did the same with my sample. Notice how my full sample is hearing half of Zhang's upper half. Additionally, children in my upper half have equivalent scores to children in Zhang's lower half. Now, what does this look like for conversational turns? The current sample had a mean of 600 conversational turns, while Zhang's mean was 1,000. Again, similar to our AWC findings, Zhang's sample of children are engaging with caregivers significantly more than the current sample does. In comparison to my sample, lower SES Spanish-speaking children had a higher mean adult word count. However, notice that both samples have completely overlapping ranges, meaning that there are some similarities in home language environments between these two different lower SES contexts. The third and final objective of this study was to answer the following question: how do home language environments for the current sample relate to a child's vocabulary? To answer this question and to measure child's vocabulary, I used the MacArthur-Bates Mandarin CDI, which asks primary caregivers to report on which words in an inventory of 113 the child could either say or not say. And the total number of words parent check off is the vocabulary size. The correlation showed a moderate positive relation between conversational turns and child vocabulary score, meaning that children who engage more with their caregivers had better expressive vocabulary scores. This research yielded three main findings. First, we found striking variability within the current sample for both measures of home language environments, adult word count and conversational turns. Second, there is a significant difference between rural and urban samples. I also found overlapping adult word count ranges between my sample and the lower SES Spanish-speaking sample. Finally, children in the current sample who engage with their caregivers more frequently had better vocabulary scores. This gives children more examples and more opportunities to learn how language communication works in Mandarin. Limitations to this study include our sample size of only 38 children and the differences in age in comparison studies. My research also does not differentiate adult word count between child-directed speech and overheard speech. Finally, we did not collect any information about specific parenting practices such as reading and playing. Ultimately, these results provide further evidence for the language disparities between rural and urban children and suggest the need for more research in this population. Finally, it is also a call for action to local leaders in education to improve early childhood development resources for families in rural China. I'd like to conclude by expressing my deep gratitude to my mentors, Dr. Roselle, Dr. Marchman, Dr. Weber, Alexis, and Maille, who guided and supported me unwaveringly throughout this whole project. Thank you very much.
That was great. That was great. We have a long list of participants, so raise your hand if you want to ask a question. There's a lot in there. Jennifer first, and then Jamie.
Laura, thank you. That was a beautifully presented summary of your research. Would you please talk a little bit about what languages you functioned in order to be able to conduct this research?
Great question. I functioned primarily in Mandarin. I worked with colleagues both at the Rural Education Action Program at Stanford and our local partners in China. I would go to families with my colleagues who were fluent who were fluent in Chinese and also Chinese themselves, and they would conduct the interviews while I would sort of serve as a observer, since it it's best for someone who's fluent in Mandarin to engage with families.
Thank you. Wonderful.
And Jamie.
Well, that was great. So two quick questions. One is, were there dialectic differences between the different areas that you studied? And the second part is, do you think that there would be differences between, say, the Spanish comparator group that you were looking at and the Chinese based on language structure and length of words?
That's a good question. So as for the dialects, the dialect in different counties varies a little bit, but Shanxi province generally everything is based off of Mandarin Chinese, and the dialect will differ a little bit, but the recorder is basically able to pick up a fairly accurate count of those adult words, regardless of dialect. And your second question was how the words in Spanish and Mandarin differ?
Right, so the structure of the language and how that might be used and might result in some differences between the groups.
Yeah, that's a great question. So there have been a lot of validation studies using this instrument, meaning that researchers have gone in and had the automated count of adult words and have compared that to inter-rater reliability. And so this has been done for both Mandarin and Chinese, and those correlations might be slightly off, but both of them are above sort of a correlation value of 0.7, meaning that both are fairly accurate, not exact, but fairly accurate measures of a home language environment.
Great, thank you.
Okay, and a real quick question from Scott.
I'll make it very quick.
Okay.
I have lots of questions, and it was a fantastic presentation. So you went into these 38 households, and as we know, there's lots of this phenomena called left behind children, where some of these children are being raised by their grandmothers and their parents are off in the city working, and others are being raised by mother or home. Is there, just from your own feeling, is that a big source of difference here? Or I don't want to ask you what is, because I think that's our next project, Laura.
Yeah, I mean, so basically 3 out of 4 caregivers in this sample were mothers, and the rest were usually either fathers or paternal grandparents. And I would say that it is typical for, my guess is that mothers will engage more with a child. Grandmothers usually have other priorities such as cooking and cleaning, and when the mother is there, usually the grandmother is as well. And so there's an additional adult to be conversing and engaging with the child. So in this pattern of left behind children, where parents migrate to more urban areas and to seek out job opportunities, the child is left with one less adult to engage with them linguistically.
Okay, fantastic. We're going to move to our last speaker in this session, Mailo Numazu, and I'm going to share my screen.
women and are a common symptom of pregnancy. Lastly, research so far has failed to conclude the true prevalence of either disorder within the female. And for the past year, I have been conducting research on the relationship between insomnia and postpartum depression in active duty female soldiers. But before I dive into what my research produced, I want to explain why I chose to do this project in the first place. So, why am I studying active duty female soldiers? Women currently comprise 15% of the total US active soldier population, and a majority of them are of childbearing age, which entails that they have unique health needs. The proportion of women in the military is continuing to rise, and despite their important role in the functioning of our military, there is a surprising lack of research regarding their health, especially when it comes to mental health disorders, such as insomnia and postpartum depression. Insomnia is a sleeping disorder characterized by sleep issues such as difficulty falling asleep and difficulty staying asleep. It's commonly experienced by active duty military when returning from deployment, and because of the unique experiences and stressors faced during deployment, military personnel are suspected to be at higher risk. Postpartum depression, or PPD, is a mood disorder characterized by depressed mood, fatigue, difficulty concentrating, and sometimes suicidal ideation. However, what makes PPD unique from other depressive disorders is that it typically arises within the first year after a woman has delivered a baby. Epidemiological studies show that about 10 to 15% of civilian women will experience PPD. Now, the similarities between these two disorders are the following. Both cause those affected to have decreased performance during their daily activities. Additionally, epidemiological studies have shown that they commonly affect women and are a common symptom of pregnancy. Lastly, research so far has failed to conclude the true prevalence of either disorder within the female soldier population. With all these similarities in mind, how are these two related? There has been a substantial amount of research on the relationship between these disorders. Growing evidence shows that insomnia increases the risk of general depression. For PPD, though, only an association with insomnia has been established, with no directionality. But there was one study that suggests a bidirectional relationship exists between insomnia and PPD, but that was in civilian women. So this is where my study comes in. My study aims to answer the questions, does insomnia before or after delivery predict for PPD? And does PPD predict for insomnia after delivery? To answer these questions, I use the Stanford Military Data Repository, which contains data for all US Army soldiers between the years of 2011 and 2014. And from this data set, I identified 10,880 women to use for my analysis. My analysis included both descriptive, to assess the prevalence of both disorders within my study population, and logistic regression models to test the associations between insomnia and PPD. Before getting into the results of these analyses, I'd like to explain how I acquired my study population. I had 3 eligibility criteria for the women that were included in my final analysis. First, they had to have delivered a baby sometime during the dataset between 2011 and 2014. Next, they had to have a minimum of 3 months data prior to the month of delivery, aka the last trimester of their pregnancy, and they had to have a minimum of 12 months data following, which totaled up to 16 months. Finally, if these women had multiple births throughout the data set, then only data surrounding their first delivery was kept. And after all this, I had 10,880 active duty female soldiers to use in my analysis. If anyone is interested in their demographics, I'd be happy to discuss these in more detail during the Q&A. With that covered, let's dive into the prevalence of insomnia and PPD within these female soldiers. For insomnia, about 10% of the women in the study were diagnosed with a sleeping disorder sometime during the 16 months of data I gathered on them. 368 of those women with insomnia were diagnosed during the 3 months before delivery, but most of them were diagnosed during the year after. Meanwhile, 17% of the study population was diagnosed with PPD. Now, for the main part of my study, was insomnia predictive of PPD? Yes. Insomnia before and after delivery, though it had to be diagnosed before PPD, predicted for postpartum depression. When adjusting for different traits of the population, insomnia before delivery increased the risk of PPD by 46%, and insomnia after delivery increased the risk by 2-fold. So this relationship was found to be significant, but was PPD predictive of insomnia? Again, yes. PPD did significantly predict for insomnia after delivery, and it did so by increasing the risk by 3-fold. However, while these disorders increased the risk of each other, there were factors that decreased their risk. For PPD, these military officer rankings which for more familiar names include lieutenant, captain, and colonel, actually decrease the risk of postpartum depression by 50% or more. And the same was true when predicting for insomnia. And there are reasons for why these officer rankings decrease the risk. The first is called the healthy warrior effect, where only healthy officers are active duty, which means they are probably already at a decreased risk for a host of other health issues. The second can be chalked up to the mental health stigmatization in the military, where these officers may have either of these disorders, but they don't want to report it to their clinician in fear that having these disorders on their chart may negatively impact their career progression. In conclusion, in my study, I found that insomnia before and after delivery predicted for and increased the risk of PPD. And vice versa, I found that PPD significantly predicted for and increased the risk of insomnia after delivery. From establishing that both of these relationships exist, I can then conclude that insomnia and PPD have a bidirectional relationship in the active duty female soldier population. These findings are really exciting because of the implications they have on this field of research. As far as I'm aware, my study is the first large-scale data-driven study on this topic of insomnia and PPD. My study also seems to be the first to address these disorders in a specific population of active duty female soldiers. And if I could find a significant relationship in this population, then maybe more studies should be done to determine if similar relationship exists in civilian populations. By having these traits, my study increases our knowledge of, obviously, the relationship between insomnia and PPD, since previous studies had only found an association, and only one small study suggested that there might be some directionality. And all of those were in the civilian population. It also increases our knowledge of the prevalence of these disorders in the military population. And lastly, and maybe most importantly, it increases our knowledge of the health needs of the pregnant military population, which could prompt further action on their healthcare. Closing thoughts. I'm very excited that my study found a significant bidirectional relationship between insomnia and PPD in the female soldier population, as it adds data to a research question that hadn't yet been answered. And with this finding, I hope my research will inspire the US military to improve screening and treatment of these disorders to help female soldiers perform at their best. I'd like to say thank you to all the wonderful advisors who'd helped me on this project. And with that, I'd be glad to answer any of your questions. Thank you.
Sorry for that little technical fail there.
All good. I was prepped to do it live, so I'm glad it worked.
Okay, yeah. All right. So let's just go straight to questions. I'm seeing a lot of clapping. Oh, okay, here we go. I'm gonna start with Jesus.
Hi, Milo. I was wondering, in your conclusions, you mentioned how this kind of research on this particular group of people, namely women in the military, hasn't been done before. And I was wondering if in your research you found any reason as to why that was, or if this just seemed to be like an oversight from the community.
The reasoning that I have found from my literature review is that for most of the history of the military existing, it has been a majority of men, and therefore research has really only had to focus on the male population. And it wasn't until somewhat in our recent history that women were able to integrate more into the military, play a larger role in its functioning. And I think that research is just taking a slower amount of time to catch up to the proportion of women that are in the military and to focus more of their research on the specific health needs of that population.
Thank you.
Thanks, Jesus.
Great, Lianne.
Okay, a question for you, Mailo. Great job with your presentation.
Thank you.
Okay, so this challenge that I brought up in your draft of sleep problems being a manifestation of postpartum depression. So how do we know these are distinct entities versus being, the insomnia being a early or subsequent manifestation of PPD?
Unfortunately, I don't think that the identification process of either disorder was specific enough to identify whether or not insomnia was misdiagnosed, or maybe PPD was just a continuation of a chronic depression disorder.
for other people not as aware of my research as my advisor, Lianne here, insomnia is a common symptom of postpartum depression. It's one of the cardinal symptoms, but insomnia itself is like a individual disorder. But for my research, I wasn't able to really distinguish whether or not when a clinician was diagnosing insomnia, were they actually diagnosing insomnia as an individual sleep disorder, or maybe they were misdiagnosing PPD for its insomniac symptoms. So future research, I think it would be really great to find a way to just like really dig in and distinguish between those two. But unfortunately, the data that I had was not able to.
Thank you.
Thanks, Lianne.
I think we have time for one quick question from Angela.
Hi, Milo. Thanks for an awesome presentation. In your presentation, you managed a little bit, you mentioned the demographics of the population. I was wondering if you could talk a bit more about it.
Sorry, I just saw people's backgrounds. Yes, I would love to talk about the demographics of my population. Just let me pull it up real quick and I will share it with everyone. Okay. Put it all the way at the end, so I have to like click through it. All right, here we go, share screen. So my study population, majority of the women were younger than 36 years of age, which I think makes sense as most women are having babies before then. Majority of them were white, next followed by the next biggest proportion of women being of African-American background, and a majority of them were had only received a high school education. Those are the like most prominent demographic features of my study population that really define the norm in like the military population. Yes.
Amazing, thank you.
Thanks, Angela.
Okay, great. So I want to take, before we get to our last session, I just want to take about a 5-minute break to try to keep us close to time. So that would be 3:47, 3:48. So we'll see you back here.
Thanks.
Okay, everybody, welcome back. This is our last session of today. We've got 4 speakers coming up. And again, for anybody who has just joined us, when we get to the Q&A part, we'll have each of the students live and ready to answer your questions. And you can just reveal the participants panel on the right side of your screen and raise your blue hand. And then I'll be able to unmute you and call on you. Okay, so we're gonna start things right off with Jacob.
Hi, everyone. My name is Jacob Thoenen, and my thesis was on non-contrast MRI of synovitis and quantitative assessment of inflammatory activity in the knee. Some quick background: osteoarthritis, or OA, is a very painful disease of synovial joints that affects millions of US adults. Synovitis is inflammation of the synovial membrane that overlays joint fluid, and this is thought to play a key role in disease progression of OA. Additionally, when synovitis is present in OA, it can be treated differently than classical osteoarthritis. So currently, synovitis is assessed using dynamic contrast enhanced, or DCE MRI, with a gadolinium-based contrast agent. As you can see on the left, the synovium can be pretty clearly seen over here, overlaying this joint fluid. However, there are some challenges with DCE imaging. It is not commonly included in clinical imaging sequences due to the increased time, complexity, and cost. Additionally, patients with decreased renal function are contraindicated from receiving gadolinium-based contrast due to the risk for developing nephrogenic systemic fibrosis, or NSF. Lastly, some recent evidence has shown that repeat injections of gadolinium can form deposits in the brain. And while these deposits are of unknown clinical consequence, it's probably best to avoid them if possible. There are 2 aims of this study. The first was to look at if the double echo and steady state, or DES sequence, could replace DCE for detection of synovitis. And the second was to look at how metabolic uptake in inflamed synovial regions is associated with the severity of synovitis that is seen on DCE MRI. So for this first part of the study, we scanned 11 subjects bilaterally for a total of 22 knees. We scanned them with 2 different DES sequences, one with a high diffusion gradient and one with a low diffusion gradient, in addition to a gold standard post-contrast DCE sequence. After we acquired these images, I created these DES hybrid images using a linear combination of the 2 DES echoes. As you can see in this first echo, both the synovium and joint fluid show up relatively bright. On the second echo, only the joint fluid will show up bright. So if you multiply the second echo by a weighting factor beta and subtract it from the first image, you end up with this hybrid image where the joint fluid is nulled and surrounded by the synovium, which is still bright. So after creation of these images, the DES and DCE images were randomized and blinded to all clinical data, and they were put into 3 different sets to be read by radiologists. So we recruited 4 radiologists to grade the images for the overall impression of synovitis, their diagnostic confidence in that overall impression, and the degree of synovitis at 4 different regional sites. And these were all graded on the 4-point scale shown to the right. These are the 4 regions that we assessed. On the left is a sagittal view of the knee that shows the suprapatellar region and the intercondylar notch regions. And on the right is an axial reformat of the same knee that shows the medial and lateral parapatellar recesses. To look at agreement between DES and DCE, we used a linearly weighted Cohen's kappa that ranges from -1 to +1. What's worth noting in this table is that both DES high and DES low showed significant fair agreement with DCE for the overall impression, and they also showed pretty similar ratings for the 4 regions, with the exception mainly of the lateral recess shown here. We also then split the readers into 2 different groups, one consisting of readers who had experience reading the DES sequence, and one consisting of a single reader who had not seen DES images before. When we looked at these, the second reader group who had seen DES images tended to do a little bit better than all of the readers combined. They showed still fair to now moderate agreement with DCE, whereas for the third reader group, this reader showed pretty poor agreement with DCE for both the overall impression and all 4 regions. We also looked at diagnostic confidence. Unsurprisingly, DCE showed mainly high diagnostic confidence with some moderate confidence. DES low tended to have a 50-50 split between high and moderate diagnostic confidence, while DES high showed mainly moderate diagnostic confidence with some low confidence and a little bit of high confidence. That's worth noting that you don't need to only have high diagnostic confidence. Having something that has moderate to high diagnostic confidence is still sufficient. So something that we noticed was that when readers tended to disagree, they had a bias to grade the DES images lower lower severity of synovitis than the DCE images. So this is an example where they would have graded them similarly. As you can see, the synovium seen on the DCE is pretty similar to that seen on DES low. It enhances similarly and it shows similar synovial volume. But in the second case, the synovitis seen on DCE has enhanced more robustly and shows a larger synovial volume than is seen on DES low. So readers would grade the DES image lower for synovitis than they would grade the DCE image. And this could be partially explained due to the fact that DCE tends to overestimate synovial volume, and that's also severity of synovitis. So this may represent just a bias in the two images and how they represent the degree of synovitis. It's also worth noting that while the DES images tended to underestimate synovitis, they still showed that synovitis was present. They just showed it at a lower severity than was often seen on DCE images. So for the second part of the study, we were planning on scanning 11 subjects. We got 5 of them scanned before we had to close due to COVID, and one of these subjects was excluded due to high pre-scan blood glucose. This slide shows this parameter called K-trans, which is a measure of permeability seen with MR. And you can basically think of it as a measure of severity of synovitis. So if you look at this region of synovial inflammation seen on the post-contrast DCE image, it also shows high K-trans values as you would expect. And if you look at the same region with a PET SUV map overlaid, you see that this region of synovial inflammation also shows very high metabolic uptake. So knowing that, it's not too surprising that when we look at the correlation between SUV max and K-trans, they showed significant moderate to high correlation for the sum of all regions. And when we looked at the regional analyses, even with very low sample sizes, there was still a significant high correlation between, or strong correlation, sorry, between SUV max and K-trans for both Hoffa's fat pad in the lateral recess. There were also positive correlations seen in other regions, but they were not significant, likely due to low sample size. So in conclusion, DES tended to show fair to moderate agreement with DCE images, but it tends to underestimate the severity of synovitis compared to DCE. As we talked about earlier, one explanation for this is that DCE is known to overestimate synovial volume. So this may just be due to a bias of the actual degree of synovitis between the two sequences. Additionally, the reader group consisting of the inexperienced DES reader showed much poorer agreement with DCE when compared to the reader group consisting of experienced DES readers. So this shows that we would need more robust training for readers who do not have experience with the DES sequence if this is to be implemented clinically. We also, as we just saw, showed the levels of glucose uptake in the inflamed synovium showed significant positive correlation with the intensity of synovitis seen using MR parameters. And lastly, I just want to thank everyone at the joint and IMF groups for all of their patience and help in making this project happen. Thank you.
Great job. Good job. Okay, so anybody have questions? Go ahead and raise your hand. Lianne has a question.
Yeah.
Hey, great presentation. Here's my question. It sounded, I know you're using the DCE, if that's right, as the gold standard, but it kind of sounded as if you might be thinking that this new approach might actually be getting closer to the truth potentially if the DCE has its own sort of built-in, for want of a better word, false positive rate maybe. Is that right? Did I understand you correctly?
Yeah, yeah. And I don't know if it would necessarily be false positive, but it overestimates the severity because of leakage of the contrast agent into the joint fluid. But yeah, we don't really know if this is necessarily closer or if it's closer to the true degree of synovitis than DCE is. I think one way we could maybe look at that is to scan patients with DES and compare that to arthroscopic findings surgically. That's one way that we can maybe look at that, hopefully in the future. But yeah, hopefully, but I'm not really sure to tell you the truth.
Yeah, no, that makes sense. So kind of an additional source of validation, essentially.
Yeah,
that would be great. Thanks.
Yeah.
I have a very trivial question, which is, has it ever occurred to you to name a band or a coffee shop Hoffa's Fat Pad?
That is not. That's a good idea, though. If I ever open one of those, I'll have to steal that.
Any other questions for Jacob? All right, great job.
Thank you.
Thank you so much. Okay, we're going to move on to the next speaker, Brandon Bergsneider. And
hi, my name is Brandon, and the title of my thesis is How Are Neurons Assembled? Exploring the Molecular Mechanisms of Nematin, a novel microtubule organizing protein. My research deals with microtubule dynamics, and this topic is important because microtubule disruption is a hallmark of several neurodegenerative and neurodevelopmental diseases. I'm particularly focused on the neurodevelopmental disorder microcephaly, which is a condition characterized by a small head and often impaired cognitive development. It is caused by abnormal neuron generation and migration, processes that rely on microtubules. Microcephaly occurs in up to 150 out of every 100,000 births worldwide, and it has no cure. Neurodegenerative diseases are related, and many of them, including Alzheimer's, are also characterized by microtubule dysregulation. So why are microtubules important in these diseases? Well, neurons rely on them to control essential cell functions such as axonal transport, neuronal polarity, and synaptic activity. Thus, it makes sense that disruption of this microtubule network causes abnormal neuron organization and function and ultimately leads to disease. However, we still know very little about how microtubules are organized in neurons. To fill this gap, I researched a newly discovered protein named nematin. Nematin was first characterized by our lab in 2012, and we know that it is enriched in neurons and interacts with microtubule-associated proteins. Furthermore, its known structural domains, which are shown here, are known to play key roles in neurological disorders such as lissencephaly and Parkinson's. However, before the research done in my thesis, we didn't know much more about it. To fill this knowledge gap, my thesis had three main goals: explore nematin's role in early brain development, understand its role in neurodevelopmental disease, and determine its molecular mechanisms. Because I have such limited time, I'm now just going to run through some of my most significant findings. First, conditional knockout of nematin is perinatal lethal and results in a microcephaly-like phenotype. Our lab previously generated a conditional knockout mouse line in which nematin expression was knocked out as early as 12 days into embryogenesis. The design of this conditional knockout is shown in panel A. For panels B and C, I want you to look at the bars in the far right of both graphs. So this bar compared to this bar. Those bars represent mice with complete nematin knockout. The rest don't have complete knockout. As you can see, nematin knockout mice were present as embryos, but did not survive until birth. This strongly suggests that nematin is crucial for survival. Furthermore, as you can see in panels D and E, conditional knockout mice exhibited reduced head and brain size compared to controls. This condition is a hallmark of microcephaly. So next, I was interested in why conditional knockout of nematin yields such drastic phenotypes. So I looked at where nematin is expressed in cells. I found that nematin localizes to sites of acentrosomal microtubule nucleation. So what that means is, if we go back to this image I showed earlier, microtubules are composed of alpha-beta tubulin dimers, and they are put together from the minus end to the plus end in a process called nucleation. This occurs at microtubule organizing centers, the most common of which is the centrosome, However, microtubule nucleation can occur away from the centrosome, aka acentrosomally, and this process is crucial for neuronal microtubule organization in particular. So I found that nemetin localizes to these acentrosomal sites by transfecting COS7 cells with nemetin in red, so here, and EB3 in cyan, so here. EB3 is a protein that marks microtubule plus ends. Thus, the direction you see the EB3 comment going is the direction the microtubule is growing. These nemetin puncta were found at acentrosomal sites, and as you can see, the microtubules are growing out of these acentrosomal nemetin sites. CAMSAT2 is a well-characterized acentrosomal minus end stabilizing protein, so I hypothesized that nemetin interacts with CAMSAT2. As panel A shows, by infecting mouse neurons with nemetin in green and staining for CAMSAT2 in red, I found that nemetin and CAMSAT2 indeed co-localize in neurons. Next, I wanted more definitive evidence of their interaction. Panel B shows a yeast-2 hybrid screen. How this experiment works is you express two proteins in yeast, in this case, parts of nemetin and parts of CAMSAT2. And if the two proteins directly interact, they facilitate expression of necessary nutrients, allowing for colonies to grow. As you can see by the colony growth in section 3 here, I found that nemetin's N-terminus, it directly interacts with a small region in the middle of CAMSAT2. Panel C shows a co-immunoprecipitation conducted in neurons. Co-IPs work by using target-specific antibodies to indirectly pull down proteins that interact with your target protein. The IP antibody is for your protein of interest, and anything that shows up from a blot antibody interacts with your protein of interest. As you can see in the top right corner of this figure, nemetin and CAMSAT2 are both capable of pulling down each other. Now that I knew nemetin and CAMSAT2 interacted, I wanted to implicate them in the same molecular pathway. Other groups have shown that when CAMSAT2 is knocked down in neurons using siRNA, neurons exhibit decreased dendritic length and less dendritic branches. So I hypothesized that if nemetin helps CAMSAT2 stabilize acentrosomal minus ends, knocking down nemetin would lead to a similar phenotype. Indeed, this is what I found. In panel A, I want you to look at the TUJ1 column. TUJ1 is a neuronal microtubule marker, and this figure shows that when nemetin and CAMSAT2 are knocked down using siRNA, there is less microtubule complexity. Panels B and C show that nemetin and CAMSAT2 knockdown neurons both had significantly fewer primary neurites and shorter primary neurite length. This strongly suggests that nemetin is crucial for proper neuron morphology and that nemetin and CAMSAT2 operate in the same pathway. Using all of the evidence from my thesis, I propose the following relationship between nemetin and CAMSAT2. When a new microtubule minus end is generated, I hypothesize that nemetin recruits CAMSAT to stabilize microtubule minus ends. Because nemetin is necessary for proper microtubule minus end stabilization, this would explain why brain development is impaired and mice are born with microcephaly-like phenotypes when nemetin is knocked out. In summary, in this thesis, I have shown that nemetin is crucial for neuronal development, is implicated in microcephaly, plays a role in acentrosomal microtubule nucleation, and interacts with the minus end stabilizing protein CAMSAT2 to facilitate proper microtubule organization in neurons. Thank you for listening, and I'd especially like to thank my readers, as well as the entire Yang Lab for their help throughout this thesis. Thank you.
Fantastic. Great job. So, questions for Brandon? Paul Fisher.
Hey, nice job, Brandon. Sorry here. What a nice painting. So in your work there, so it looks like all the models were knockout models. Do you have any idea, sort of we were talking about in neurodegeneration where it may not be an all or nothing, but it may be an abnormal protein is the other part. Do you have anything that in the model that approximates more just a present protein, but one that's dysfunctional?
That's a great question. Our current models don't account for that. The mouse models are knockout models, and the cell culture we use siRNA, so that's knockdown models. But certainly that is a topic that deserves further exploration. And I think no GWAS studies have yet discovered nemetin's implication in neurodegenerative diseases, but maybe through those sorts of studies, we can determine certain types of mutations in nemetin and potentially dysregulations that might be implicated in disease. But that's, yeah, great question.
Annette.
Thanks for a great presentation. I was curious about the nemetin and CAM-AP2 interaction at the biochemical level. So you have an idea of the function, but do you know how those are interacting at a more detailed molecular level?
Yeah, that's a great question. And let me actually see if I can share my screen to help answer that. So are you able to see my presentation?
Yes.
Okay. So after we discovered in our lab, the interaction between CAM-SAT2, I was interested in analyzing which specific domains of nemetin and CAM-SAT2 are binding to each other. So I conducted domain analysis and cloned different regions of nemetin and different regions of CAM-SAT2 and conducted a few experiments. I conducted the colocalization experiments to see if they colocalized, and also the yeast-2 hybrid experiment. And this slide here is a overview of what we know about nemetin and CAM-SAT2. And so if you look at this region here, we know that nemetin's N domain is what is sufficient and necessary for CAM-SAT2 binding. And we know that this region in the middle of CAM-SAT2 is necessary and sufficient for nemetin binding. We don't know the exact structural dynamics between those two regions, and so that's definitely an area of further study. And I'd also like to further narrow down on those specific domains and make them more specific. But yeah, great question.
Great, thanks.
Okay, that was great. So if there are no questions, we're going to move on to the next presentation, which is Maimi Higuchi.
Currently, we see an increasing gap between organ donors and patients. Meanwhile, using human stem cells in animal models is ethically limited, even though they can make more accurate models for disease and research. Hence, one important goal is to make organs for people. We have mainly two ways to accomplish this, in vivo and in vitro, but seeing as in vitro is highly limited and lacks interactions with surrounding organs, we focus on in vivo. With in vivo, a method to generate these interspecies chimeras involves injecting cells into embryos that lack the ability to form a particular organ by knockout. Cells then contribute to the organ where it would normally form. However, a key ethical issue is that cells later contribute to tissues outside the desired target organ, such as the brain or reproductive system, where we don't want human cells to be. Hence, current studies focus on cells that are more restricted in development than pluripotent stem cells called progenitor cells. For proof of concept, in this study we injected mouse definitive endoderm progenitor cells into mouse embryos, which should only contribute to endodermal lineages during development. So the purpose of this research study is to overcome limits of the fact that we are injecting differentiated definitive endoderm progenitors in early stage embryos. The overarching long-term goal is to ultimately generate human organs and human livestock chimeras so that human cells only contribute to the organs of interest and nowhere else. The first stage involves generating mouse embryonic stem cell or MESC lines with 3 transgenic modifications and labeling: inducible BCL2, a SOX17 reporter, and constitutive TD tomato expression for cell tracking. Previous studies state that overexpression of B-cell lymphoma 2, or BCL2, which is a gene that inhibits apoptosis, helps cells contribute to early chimera formation even with differentiated progenitors. SOX17 is a transcription factor expressed in only endoderm tissue. So with GFP attached to SOX17, all these progenitors are green, which allows me to pick them. We then optimize the protocol for converting MESCs to these progenitors in the second stage. In the third stage, we sorted for these progenitors, injected and analyzed them. If chimerism was significant, we plan to let embryos go to full-term birth. Here are our findings and interesting points. Initially, we received MBSC line SOC17 GFB1. However, for this study, healthy cell lines are important because abnormalities may contribute to cell variations and differentiation, which makes it difficult to accurately analyze later cell survival. Unfortunately, as we see in A, the cell line tested mycoplasma positive in comparison to its negative control. In addition, in B, the cell line gained an extra chromosome 6. Therefore, we made new cell lines containing the same markers and called them SUN 109 and 110, shown in B and C respectively as they glow TD tomato red. While they were all mycoplasma negative in E, SUN 110 also had abnormalities where it is missing a Y chromosome shown in A. Interestingly, we qualitatively noted that 110 was prone to random differentiation in regular cell culture as seen in D, where we see more flattened cell colonies than typical round colonies. This may support current literature that abnormalities could cause aberrant changes in DNA makeup and development paths. Going forward, we then only used 109 and chose subclone 109.1. In F, we further confirmed that the BSCL2 construct was truly working on 109.1 through successful cell contribution shown in red. Then, heading into the next stage, we optimized because the original endoderm protocol was for human cells. The first differentiation media used is EpiLC. As seen in D and E, 3 days in EpiLC media seem to significantly increase percentage of cells expressing GFP, a proxy for the progenitors. A reasonable interpretation for 3 days is that cells sat in media longer compared to 2 days. However, as to why 3 days is better than 4, cell death may be the cause. It's possible some chemicals in EpiLC are toxic, as we see less cell survival the longer the time in A through C qualitatively and E quantitatively, but more investigation is needed. Note A through C shows cells in 2, 3, and 4 days of EpiLC respectively. Hence, overall, we interpret that the 3 days in EpiLC media may be the best balance between cell survival and greater differentiation. We further optimized for starting cell density and matrix type, which showed significant conversion to definitive endoderm progenitors at 5K per centimeter squared and mouse embryonic fibroblasts or MEFs. Once optimized, we see that these cells successfully express GFP over time in C through G. Furthermore, previous studies state that definitive endoderm progenitors are likely PDGFR alpha negative and CXCR4 positive. In stating for these in H, all GFP positive cells were CXCR4 positive and PDGFR alpha negative, which suggests GFP positive cells were all definitive endoderm. As we injected sorted definitive endoderm progenitors into mouse embryos, they died very quickly, so we investigated chemicals that could support cell survival and growth. For example, Rho kinase or ROCK inhibitor is known to promote cell survival in human stem cells after single-cell dissociation and other stem cell protocols. Insulin is also a known growth factor added to nearly all cell culture systems, but it's absent from the embryo media we used. Hence, we tested effects of ROCK inhibitor as TCS and insulin as ITSX. In A, on the left, we use 100 nanomolar ROCK inhibitor, and on the right, no inhibitor. Clearly, the inhibitor contributed to short-term survival of the progenitors, shown in increased fluorescence. However, they died quickly afterwards. This is also supported in B. Yet in B, increasing the concentration of ROCK inhibitor to 800 nanomolar appeared to increase cell survival significantly. Meanwhile, the effect of ITSX seemed to depend on ROCK inhibitor. In C, with the absence of ROCK inhibitor, ITSX did not appear to have a significant effect on cell survival, as most cells died. In contrast, with the presence of ROCK inhibitor, ITSX increased cell proliferation more significantly compared to just using ROCK inhibitor. However, concentrations of this inhibitor impacted cell survival and embryonic development. With D, we see that a lower inhibitor concentration had no cell survival after live imaging, but had normal embryonic development. And further support with F, E 9.5 analysis of these embryos had no cell contribution. In contrast, in E, higher concentration had increased cell survival, but delayed embryonic development. Embryos died by E 9.5 as further support of this observation. Thus, we found significant patterns that explore challenges for specific differentiation and successful development. Mycoplasma and chromosomal abnormalities that occur frequently may impact differentiation developmental pathways. Balance is needed to prevent high cell death and maximum conversion to these progenitors, which occurred with the following 1, 2, and 3. Furthermore, enrichment of the embryonic microenvironment with ROCK inhibitor and ITSX resulted in great improvements in definitive endoderm progenitor survival and proliferation. Thus, ROCK inhibitor and ITSX likely play significant roles toward our goal of generating lineage-restricted chimeras.
Thank you. Thank you so much. Yeah. Okay, so anybody who has a question can raise a hand. I'm gonna defer to others for just a second. Okay, I'll real quick ask my question. I kind of wanted to go back to the very basics. So the way you started your presentation, you identified a really significant problem with the way chimeras are produced right now. Given, and at the end, you provided ideas for maybe trying to optimize this. How close do you think we are to being able to overcome some of these hurdles?
Okay, that's a really good question. Okay, I'll try to share my screen here. There we go. Essentially, back to the basics in my paper here. So the initial ethical problem was that we were, the overarching large goal was maybe we can inject human stem cells into animal embryos and have them grow later on, and then hopefully have human cells grow into an organ inside an animal, later harvest that human organ for use into patients. But obviously ethical problems are that those human pluripotent stem cells would contribute everywhere to the animal, including the animal brain and then the animal reproductive system, and that has major ethical consequences, so we don't want that to occur. So essentially, with my study, I try to restrict that to progenitors, which are further developed from and restricted compared to pluripotent stem cells. And so there were a lot of problems with optimizing the protocol and just seeing like different concentrations, different matrices. I think it's just that it's such a relatively new field in terms of making interspecies chimeras that it's just a lot of steps just with the simple optimization. And so there are quite a few. So this, we went here with optimization and then just different bacterial possible consequences and just different supplements. So I think we're still in the beginning stages in terms of achieving this long-term goal. But the field is rapidly changing even within the past few months, and so it's a very exciting time to be in this field.
Sorry, I was muted because you can hear Caltrain running right by my house. That is really exciting. Thank you. Thank you. Okay, if nobody else has questions, I'm just going to move to our final speaker of today, Angela, and let me share my screen.
Everyone, my name is Angela, and today I'll be presenting my honors thesis project, Identifying Sex Differences in Placental Inflammatory Pathways. So first, a bit of an overview. Pregnancies with a male fetus carry higher risk of complications, including higher risk of miscarriages, preterm births, and neonatal ICU hospitalizations. In these three things, inflammation is a common unifying factor. So, because inflammation in maternal circulation affects the fetus through interaction with the fetal placenta, we hypothesized that sex-dependent pregnancy outcomes could be caused by sex differences in inflammatory pathways in the placenta. And while we don't fully understand why males and females have very different developmental outcomes, we do know that males are 1, sexually dimorphic and larger than females, and 2, more allogeneic, meaning that they are more immunologically distinct from their mothers, due to the HY antigen that is expressed from the Y chromosome. So a little bit of a roadmap for my presentation. First, I'll be going into some background, methods, then continue on into results and discussion and conclusions and next steps. So first, a little bit of background. What is the placenta? So the placenta is a multifunctional tissue that develops during pregnancy and serves as the interface between the mother and fetus. Importantly, it is a tissue of fetal origin and shares the sex of the baby. For this project, we chose to look at the chorionic villi, which is shown enlarged on the right here. We chose to focus on this structure because the chorionic villi is the most important location for maternal exchange, as they are bathed directly in maternal blood. So the structure of the villus consists of a bilayer of trophoblast cells. On the outside, you have a multinucleated syncytium tissue, and on the inside, a layer of cytotrophoblasts. Finally, in the stromal core, there are immune cells, fetal endothelium cells, as well as fibroblast cells. Our previous studies show that some autosomal genes are differentially regulated by sex. In a bulk RNA-seq done between 11 females and 12 males, we saw 3 genes that were particularly interesting to us. They are LGALS13, CCRL2, and LGALS14. Now I'll go into the details of their functions. So first, LGALS13 and LGALS14 are placenta-specific proteins that are known to induce T-cell apoptosis. Of the two, LGALS13 is by far the better known and is also known as placenta protein 13. It has been suggested to be a biomarker for preeclampsia. Additionally, previous studies have shown that in first trimester miscarriage CV samples, there is a decreased abundance in both of these proteins. So on the left here, we see a control stain of LGALS14, and also on the right, we see the same protein stained in a miscarriage sample, and you can see the large decrease in abundance. CCRL2 is the other target gene we are focusing on. This is an atypical chemokine receptor, meaning that it is a transmembrane protein, but it does not signal through G proteins. However, we do know that CCRL2 is expressed in barrier cells, including epithelial and endothelial cells, as well as in leukocytes. However, we don't know about its expression in the placenta. Other members of the atypical chemokine receptor family are known to be expressed in placental syncytium, and so perhaps CCRL2 is also expressed in the syncytium as well as in fetal immune cells. Additionally, we know that CCRL2 is important for immune cell recruitment, and studies have shown that CCRL2 is required for CXCR2 dependent neutrophil recruitment. So perhaps it is playing this role in the placenta as well. So all this leads to my 2 questions. 1, does elevated transcription translate directly to protein abundance? And do the same differences we see in our RNA-seq translate to protein? And finally, what is the localization and expression pattern of our genes in placenta? So moving into methods. The original research design for this study first consisted of gathering a research cohort. So my cohort consisted of 9 females and 11 males, ranging from 11 to 16 weeks of gestational age. Then practice stains and optimization of staining were performed to ensure that my methodology was sound. Next, I moved on to serial staining of my cohort samples so that I could have a positive control and a negative control on the same slide as my actual protein. Then immunohistochemistry staining would occur for all of the samples, and then I would score and quantify the stainings, and finally be able to compare any sex differences between the stains. So a little bit more detail on the immunohistochemistry staining itself. The IHC, one of the first steps done was quenching of endogenous peroxidase activity and a long period of blocking to ensure a decrease in unspecific staining. Then my samples were treated with both primary and secondary antibody, and then visualized with DAB under light microscopy. So although the COVID pandemic interrupted this study midway, I still have some practice stains and some interesting findings to share with you guys. So moving on to results and discussion. This is an image of cytokeratin 7, which was our positive control and a known trophoblast marker. So we see that CK7 is intensely expressed on the syncytium and the cytotrophoblast of the villus. But we see that within the stromal core, CK7 is not present on either the fetal endothelium or any stromal cells. For LGALS13, in term placenta, we saw that it was also intensely expressed on syncytium and cytotrophoblast. Additionally, we saw that LGALS13 was present and abundant on certain stromal cells that have the morphological characteristic of immune cells. Finally, we saw that LGALS13 was shown to be present on fetal endothelium. We saw the same thing for LGALS14. We saw that it was expressed in syncytium and cytotrophoblast, in stromal cells that have morphological characteristics of immune cells, as well as in the fetal velium. For CCRL2, our findings were slightly different. So we did see that CCRL2 is present on the syncytium, but not actually on the cytotrophoblast. Additionally, in 8-week placenta, we were able to identify the microvillus structure of the placenta, but we saw that CCRL2 is not present on this structure. Finally, we saw that again, CCRL2 is present on fetal endothelium, but not as intensely, and also not in all cell types of the fetal endothelium. For immune cells, we saw that CCRL2 was present again in some stromal cells that had morphological characteristics of immune cells, as well as in cells that exhibit characteristics of maternal circulating white blood cells. Finally, in 13-week early trimester placenta, we saw that CCRL2 is present on cells that have the characteristics of neutrophils. So what does all this mean? So for LGOS 13 and 14, my findings were able to support previous findings that LGOS 13 and LGOS 14 are present on fetal endothelium. In addition, our findings help us extrapolate a bit more about the immune function of these proteins. So previously I mentioned that LGOS 13 and 14 are decreased in placentas from first trimester miscarriages. So this miscarriage could possibly be due to a lower level of T-cell apoptosis, leading to insufficient downregulation of the maternal adaptive immune system and harming the fetus. For CCRL2, for the first time we were able to characterize its staining pattern in both first trimester and term placenta tissue. So we saw it was present in fetal endothelium, as well as in syncytium in some morphologically characteristic cells that look like immune cells. Additionally, we saw that it was present in 13-week placenta, showing that CCRL2 is present in early placenta. So what is CCRL2 doing there? Well, like other receptors in this family, CCRL2 was seen on syncytium. So perhaps, like other receptors, it plays an immune cell recruiting role to recruit immune cells to these areas of the placenta. Finally, some conclusions and next steps. This was the original research design, and after doing the study, I am hoping that the next step will be to co-stain the stains so that I can more specifically identify the cells that I have preliminarily characterized here. So for example, I hope to stain the cells that I have preliminarily identified as neutrophils with Lys6 to ensure that they are neutrophils. Then I would move into scoring and quantification so that this whole study could be a little more quantifiable in nature. And this would allow me to finally understand the sex differences that I'm hypothesizing about. And finally, a huge thank you to my mentor, Dr. Braun, to the entire WIN lab and my readers, Dr. Wynn and Dr. Baker, as well as my friends and family for making this project possible. Thank you so much. And now I'll be taking any questions.
That's excellent. Great job. Great job. Okay, so we've got some hands up and I will go straight to Annette.
Hi, Angela. Thanks for a nice presentation. In your preliminary work, it sounded like you had some interesting results on the CCRL2 localization, but I wasn't sure whether that was for samples from males, females, or both.
That's a great question. So the results I presented for this study were from my practice tissues because these were the stains that I chose to optimize on and the samples I chose to optimize on before I moved into early term sex samples. And so what you're seeing here is actually a male term placenta. And that's the results that were shown for CCRL2 in this presentation.
Great, thanks.
Okay, other questions? One of the things I just wanted to point out actually how well structured your presentation was, and given that I just really liked the way that, you know, given that your work was interrupted at a very crucial point, you were able to sort of show us exactly on that map where it was interrupted. So I thought.
Thank you. I definitely got really lucky and was able to squeeze in like that very last round of staining. That was very helpful.
Little bit, yeah. Right, so you did tell us where you would go next. Let's say that you were on the verge of opening a lab to study this and you had a giant grant. what are some other things that you might do?
That's a really interesting question. So actually after this study, so this study will only show us where the proteins are present and their localization within different cell types. But I also want to delve deeper into what they're actually doing there. Because for example, CCRL2 is not very well known, and we don't really know what its function would be in the placenta. So doing some functional assays, maybe using placental cells to do like more cell-based research would be really interesting for me, and a big grant would be very helpful.
Of course. Okay, last call for other questions. Okay, so with that, let's just give everybody, not only in this session, but the entire day, a huge round of applause. Thank you to everybody. I just feel so grateful that I've had the opportunity to get to know just a tremendous group of students. So, you know, a little bit better through this process, and I really appreciate it. And I'm going to put you on gallery so that I can see everybody. There we go. Okay, sorry, don't need to see myself. Anyway, thank you all again so much, all of the mentors who were here to support students and to listen to everybody's talk and to ask really great questions. And I just want, well, I should give the students also an opportunity to applaud the faculty who are here. So let's give the faculty a big round of applause. Okay, thank you. Yeah, and I'm seeing some claps pop up too on the participants list. So thanks to everybody. And again, I wanted to just thank Jeff Gallus for his support of this symposium. And I look forward to future years when we can have a fancier version of it that is not in my dining room or wherever you all are. And thanks also to Jesse Friedenberg and Carlos Seligo for tech support, and to just everybody for taking part. So thank you all, and I'm going to draw to conclusion the 2020 Gillis Senior Symposium in Human Biology. Thank you all. Happy Friday.